Plausible probability it's going live.
We are going live. Well, we're gonna have to keep track of that guy too. Is he doing an after show on his morality debate?
I think so.
Wait, is he with Smokey for that debate?
Yeah,
he was.
Did you watch that shit?
That debate was a shit show, honestly.
Oh my god!
I had no
interest. Oh, that was a shit show.
Did you see Alpha Wolf? How he was lusting to go to an island with 4 other dudes so he could like Alpha Wolf all over their shit and like drink their water. I mean, these fucking Christians. I mean, I'm sorry. Yeah, some fucking Christians without God apparently would start killing us. I mean, is that it would be nihilism?
I interpret that as self-incrimination, but don't tell me.
I know they're both admitted to being psychopaths if they didn't believe in God.
Smokey said that?
Well, no, he didn't say that outright, but Smokey and John both said that without, some higher authority, they would just start killing shit.
I just realized we're live.
Oh, we are live.
I wanted to say shalom, brothers.
Shalom.
Shalom.
How are you doing, brother? Have you been good, brother?
Doing pretty well. So back to morality, I guess.
Morality. But I mean, I just can't. And Smokey say his shit was so naive tonight. His idea of atheism and how we're supposed to rationally decide that, well, seeing as how we just die and then there's nothing. I mean, the only thing that matters is ourselves, and we should just go after the pleasures or whatever. I mean, it's like we should.
I'm really concerned, for the types of things he probably did while he was an atheist in that case.
I know.
Jesus Christ.
Well, I think they're both. They sound like psychopaths when they talk about what it would be if they didn't believe.
I've said a lot of mean things about Maddox, and he's kind of had them coming. But man, that guy's been losing some fucking sleep. I mean, if you looked at his eyes during that debate, it just looked dead. I mean, it's like, I've had late nights before, but I mean, geez, these 9-hour streams and stuff are really taking their fucking toll. If you need help, man, or if you need to get some sleep, just, fuck off with the live stream. I mean, I thought you might have learned this in college if you graduated college, but gee whiz.
Yeah. I gotta see if he's got a live stream tonight.
Oh, who the fuck knows?
Oh, let's see here. Save him chat. I saved him chat. Let's see here. Well, guys, say something. I'm looking at something. I'm working on something.
Well, we're hoping. Oh shit, he is in the live chat. Otangelo, my main man, hop on screen. Let's chat.
Otangelo, did you get your audio fixed?
Oh, speaking of audio, I am outside because my brother's church group is having Bible study in the room where I normally talk about my godlessness.
Oh, so, go in there.
No, well,
really.
So I'm outside today. If y'all hear crickets,
I mean, I can definitely hear the crickets.
Okay.
Is there cricket fucking that's happening there? I can hear the crickets.
There's plenty of locusts around, I'm assuming.
This channel does not condone cricket fucking.
Crickets are going to do what they're going to do, okay?
Okay, no, there's no after show yet on his channel, so I don't know what's happening. Did
you send Tangelo the link?
No.
Are we gonna talk to him for some time, or what's happening here?
Are we gonna talk to Tangelo?
Are we?
Not about abiogenesis.
Let's have him talk about cells and shit.
No.
Why not?
Let's talk about evolutionary mechanisms.
No. Let's talk about morality. He says it's straw man.
Oh, actually,
let's talk about morality. That would be. Let's see where it goes. It's
because I think Otangelo has interesting philosophies with regards to dualism and stuff.
Interesting to say the least.
Yeah, I don't think it's a straw man, Otangelo. Smokey was basically saying if you're an atheist, then you have no reason to be moral or care about anybody. So,
needless to say, Smokey's mental faculties have gone a little haywire.
How am I straw manning him when he said that out loud?
How do you straw man when Smokey says something stupid?
Yeah.
Oh, by the way, when did he's correct? Wait. Okay, apparently Smokey's correct when he says there's nothing stopping me from murdering everyone.
11th dimension is here.
Hello, 11th dimension. What was I gonna say?
11th dimension. Oh, wow. He's here.
Didn't know Demeter was here.
Cool. What was I? I was gonna say something about shit. I wasn't gonna say something about shit, but shit, I forgot.
I know, it happens.
What did Jeffrey Dahmer say, Otangelo? He said if he wasn't a Christian, he would kill everybody. Jeffrey Dahmer was a psychopath, Otangelo. Are you holding him up as an example of a fine Christian fella?
Are you holding up Jeffrey Dahmer as a pristine example of Christianity? Please tell me. Oh, please. Oh, come on. Please tell me we're going here. I would. I want to see where this goes. He did convert. Oh yeah, because
Jeffrey Dahmer
almost
is
getting
the ideal
penalized.
Christian.
Oh yeah, because getting penalized with the death sentence definitely won't lead somebody to have a religious
Yeah,
conversion.
right.
Yeah.
Oh yeah, I'm sure there was nothing to why he converted to Christianity right before he died.
Yeah. Yeah. Alpha Wolf tonight was just lusting to go to the. I swear he would, if he found out there were people on stranded on an island fighting over a jug of water, he would book a flight to go there. But you know how when you watch a series where people are abandoned in some situation like that, there's always one alpha wolf, and he causes the entire drama for the entire series. Well, what I would do is I would shoot him first, in the first 5 minutes. Just fucking blow the alpha wolf away, and you won't have any drama.
Yeah.
He converted without expecting to die soon after. Really?
He was on death row.
He was on death row, Otangelo. How much more close to death do you want him to be? In the electric chair?
Was he? Does he get to go to heaven now and sit up there alongside? I'm sure Hitler converted right before he shot himself too.
Come on, Otangelo, jump in the room. I'm going to have a fun time with this conversation. Please don't talk about Jeffrey Dahmer. I've watched way too many documentaries on this, and I know way more than I should about this.
Is he the one that Zac Efron had a movie about with or something, or was that Ted Bundy?
It might have been Ted Bundy.
Ted Bundy had 8 movies or 10 movies about him. He was fascinating.
Ted Bundy was one of the strangest, probably most curious murderers there that exists.
Wasn't he a good Republican? Didn't he work for the Republican Party for a while, or was that?
He was a candidate. He wanted to get into public office. He was very active within his community and his church, though.
Mm-hmm. God, I just breathed in water. I'm pretty sure. What's that? The clown dude?
Gacy.
Yeah,
Gacy.
John Wayne Gacy. I'm pretty sure he was a very avid member of the Democratic Party or something. So
I think he was also a member of his local church and quite a respected
yeah, I'm pretty.
man.
Gacy had, for some reason, he had a bunch of people with the wool pulled over somehow. Serial killers are weird. It's crazy how
if you want to be really fascinated, look into Jim Jones.
Jim Jones. Yeah. But I mean,
God, that guy was fucking bizarre. I mean,
for me, serial killers, the craziest part is that they have families and regular lives and stuff, right? So they do this shit on the weekends, and then they just go and they're completely normal for a week. I don't understand how that mentality works.
A shit ton of drugs. In the case of Jones, Jim Jones, he was on drugs pretty much his whole life.
Yeah.
Well, Jim Jones, the founder of Jonestown?
Yeah.
Well, okay, but he was actively a cult leader, right? He wasn't nobody would look at him except for the people in the cult and be like, oh yeah, that's a regular stand-up member of society.
For a while, though, he did a very good job of concealing that it was a cult.
Yeah.
So Like, that's the thing. Like, he was politically active, and there were things, like, because he was in such a close-knit community, not many people, like, really, like, encroached on that territory or, like, felt the need, like, to get out of that community or say anything about it.
Yeah.
Although, unfortunately, people did, and that's one of the reasons he went to Africa and built his own cult compound. And
He went to Guyana, I think, in South America.
Yeah.
But yeah.
Oh, let's see here. I'm trying to find something. There's a question. Leophilius has a question. Where's the question?
I have a question for Leophilius. Why the fuck aren't you in the room?
Where's Leophilius? Wow.
Oh, you said they appreciated the thumbnail I made. Nicholas Whitmire did.
Yeah.
Oh, really? Did they?
He said that when he saw that, he realized that we were basically, I don't know, something about how we could roll with a punch, and he really thought we were cool for that.
Oh,
So
I
he
appreciate
complimented us.
Yeah.
That's good. I mean, I'm glad that Nicholas can make a statement without, like, saying something completely horrible after it.
Yeah.
He can be taught.
Well, I think Nicholas, he seems like he came in very defensive against Speed last week. But he seems like you could have like a pretty like respectful, nice conversation with him. He seems like a nice dude.
Yeah, I think he was trying to be aggressive.
Yeah, I think he was.
The thing is, though, it's just like I don't understand like why he has such an ego around him. Like, I mean, and not just, you know, like kind of like one of those things where it's like, ha ha ha, I'm smarter than you. It's like, no, I know things that you don't know. My brothers know things that you don't know. You are wrong, type of thing. And it's like, you know, man, I really want to take this seriously, but making it really, 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 really difficult.
Oh, you know, another interesting thing, though, about Nicholas Whitmire, him and Smokey, the two who are actively like, yeah, I don't really know too much about like science, are the only two that have agreed to take up Dan's challenge.
Really?
Yep.
Oh.
So all the ones who claim to be experts, the guy who took biochem at MIT, none of them are taking Dan's bio challenge.
Yeah.
I wonder why.
Really? Yeah,
you know, the people the guy who took biochem at MIT, the guy who published a paper in PLOS Biology. Yeah, these are the ones that aren't taking his challenge. I wonder why.
Yeah, it's wow. Yeah, I'm almost surprised that Smokey like is voluntarily doing that.
Well, okay,
the thing is, like, I have a certain mental image of how that's going to go out.
Uh-huh.
And it's
Well, one of the few things I have respect for Smokey for is his ability to discern what he does and doesn't know. You know what I mean? Like,
okay,
for some reason he thinks he can go into debates about evolutionary biology, but even in those debates he claims, yeah, I don't know much about this.
Okay,
right?
The issue, though, fundamentally there is that, like, even though he knows he doesn't understand, like, the type of stuff he claims to understand,
mm-hmm.
he doesn't seem to, like, say that publicly. Like,
I've heard him say it plenty of times.
Really? Okay. Maybe
yeah,
He
does it in most discussions that are about biology, right?
Has he ever done it in conversations about abio?
I don't know. No, all of the contexts I can think of it in are related to bio. So I don't know about like orgo and abiogenesis and all that kind of stuff. Yeah, Smokey's an interesting character. He actively admits that like if evolution is true or not has no effect on his theology. So I don't know what his dog in this fight is other than subscribers.
To me, that's the most confusing thing about it.
Yeah,
it's like you don't have a dog in the fight, yet you're involving yourself in this conversation for whatever reason. I don't know why you're like. Why are you like going so hard at this conversation if it's a non-essential for you? I mean, it just doesn't add up.
Yeah, it's something to do with like what he mentioned in Dan's stream yesterday about his brothers and sisters and how he feels he has to defend them. And I really think in a private talk with him, I was trying to he was talking about Maddox being some authority in genetics, and I kind of went after Maddox and with Smokey and explained to him that Maddox doesn't know what he's talking about.
Yeah.
And it was shortly after that we had our falling out, and I think that was just one more, nail in my my trash can coffin. So
Your trash clown coffin.
It's just
trash clown coffin. Yeah, coffin.
I don't know why, like, if Smokey is scared to do it or something, but it's like, I just want to hear the words, Maddox, you're wrong and you're being stupid.
Yeah.
Like, I don't expect him to say those words.
Well, I think it's the whole idea, like the same I think the reason Smokey defends them is the same reason that like they defend people like Jason Ezard or Nephilim Free.
Yep. Exactly.
It's because they see like, oh, well, he's a defender of the faith,
Yeah.
And whether he's right or wrong on specific issues doesn't make a difference to his overall theological point.
Okay,
but in my opinion, it definitely does. It gives your theological point a bad name.
Yeah, but I mean, that's kind of like the other point too. Is like, if the points they're making like makes Christianity look bad, like if you're giving Christianity, like making Christianity look bad, and you're associating with that, and number one, you can't be you can't be like triggered and offended when people try to associate you like with that type of belief structure. Like, since you hang around people who just non-stop talk about it. But the other thing is, like, if you don't think they're correct, or like if you think, like that this isn't an important topic, it doesn't make sense like to really get off like or to have these conversations so excessively. Like, it just
Jason Ezard is called a drongo.
Drongo.
I think every time I hear Ian talk about Jason Ezard, it's a different word. There was that other one that began with a G.
Yeah, he had another one. You had another one the other day.
Or something.
Yeah. So Nicholas learned something. He corrected something SFT said, who got a little irritated. They argued. I would like to hear that. I want to hear that exchange. I want to know what this is.
That's something I want to see.
Speed SFT 9-hour stream latest. Oh,
let's just do a react video to all 9 hours of that stream.
The last 4 hours was dead air.
We owe our audience something, at least. So
SFT fell asleep and everybody else left. Praise, they said, was drunk.
Wow.
Really?
No, not Praise. Yeah, Praise I am that I am. He was he frequently starts a stream and he goes and takes a shower or walks the dog or whatever.
Standing's not as bad about it as like Maddox. Yeah. Maddox just goes to sleep, comes back the next day like 13 hours later.
Many good Aussie words. Anyway,
the thing is, like, Maddox wonders why his computers are dying left and right. I wonder. Running your computer 13 hours in a row, like hosting OBS, like in several other tabs.
Yeah.
You wonder why your computer might be having a difficult time, like moving along.
Yeah. To wrap up the morality thing, basically, when you go to Walmart, if you go to Walmart, there's like 50, 100 people there. You don't expect any of them to come running at you with the knives from the cutlery section. I mean, you pretty much trust other human beings, not because they're all Christians, but because we have it built into us. That's the way we fucking behave with each other. I mean, I don't think you need to rationalize morality unless, you run into a gray area where you have to get together and powwow up a solution for a difficult problem. But
generally society wouldn't be very good at like, I almost think of it as like an evolutionary advantage, right? Because you can't have like social cohesion if people are just murdering each other for fun. Like, you can't develop complex society, and that's one of the things that's let our species be so successful. Since Neanderthals has been a pretty hot button topic recently, like they think that's one of the things that helped our species win out more or less over the Neanderthals. Not necessarily win out, but
didn't they ask about Neanderthals during the debate?
Which debate?
Whether or not,
whether or not if they were humans, and then if they weren't humans, that means they'd be put into zoos and blah 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 blah.
I like the way Smokey said it. Do you think Neanderthals were animals? It's like we're all animals, dude.
Yeah, I'm an animal. I'm a metazoan.
You're a fish.
Yeah,
I'm a fish.
I'm a primate.
I can list at least most of the classifications of animal I am. I'm not just
I'm
like
a eukaryote.
yeah. Well, I mean that's not an animal though.
No. Well, wait.
We got to learn these Aussie terms so that we can throw them at Ezard if we ever get an opportunity.
Yeah, eukaryotes don't fall into
Will he know
animalia.
what we're talking about? Known for their annoying screeching. Yeah, screaming at his dogs. Let's see. Okay, we got something from 11th. This is not about killing, it's about surviving on social species. Yeah. And people are not going to fight because fighting takes a lot of energy and you get hurt and it's risk, and mutualism is much more likely. So, yep. Anyway, so let's see. Maddox speaking. This isn't exactly him speaking in tongues, but this is an example. Leo Filius asked him a question. I didn't know Leo that when this happened, and Leo Filius asked another question. Somebody asked about ribose being found in meteors, I think, and I think we have it right here. And John answers,
assuming it's the one from last year. There's two things about that. One, okay, they may have seen some amino acids. They think it might be a protein, not 100%, but the
question was about ribose.
Oh boy.
What often gets glossed over is the fact this asteroid that they're referring to crashed into Africa, I think back in 1989, and they didn't, quote unquote, discover this protein until literally 30 years later.
Literally.
And we know full well that Erica was talking about this in terms of contamination. It's also not a completely unrealistic or implausible and improbable outcome that this is a result of contamination. So I know there's excuses on that whole deal, but
he's talking about the
either way, it doesn't really.
He's talking about the protein that was found like in the asteroid. I mean, okay.
Wait, so
what
what was the original question about ribose?
Oh, let's see here. Okay, electron transfer.
What was that
were you
question,
saying?
Michael?
Mutable?
Yeah, the thing he's talking about is like a protein that's like was found in a meteorite, and it was very strange because it's a protein that incorporates a metal into its structure.
Okay, Dapper Dino. The question is for both: how does the recent discovery of persistent ribose in asteroids impact your ideas about abiogenesis? And he did say ribose. And this, I think, is where Dr. Dan noted that.
Speaking of Dr. Dan noting that he thought ribose was a protein, he's in chat saying that, is this when John thought ribose was a protein?
Thank you, Dr. Dan. Yeah.
Yes, it is.
Yeah. This is the one. We're trying to find the one where John thought that I think phospholipids.
That's another thing I don't get about these guys. If you don't know something, it's okay to say I don't know. Like, why do they have to posture and like pretend that they know everything?
Yeah.
Because if John doesn't do that, it makes him look like an idiot, and then other people are like, oh, well, why should I listen to John if he's just
he
fucking
looks
retard?
like a bigger idiot if he thinks ribose is a protein.
Well, just let him think, okay? I mean, he can barely do that. You got to give him something, okay?
I mean, like I said, okay. I don't know. I just feel like if something comes up that I don't know about, I either shut up or I just say I don't know, right?
That's because you've been through public education and you've been humbled.
Well, not public education, but I think more just university and being around people that are smarter than me.
Yeah.
So, I go to a private university. Yeah.
Oh, okay. Wow. Okay. Yeah.
Let's see.
Excuse me.
Did Creation Myths get the link in for this?
I'm not sure if I sent it to him tonight.
Did you not send it to Otangelo?
I did not send it to Otangelo.
Oh, I thought you did. Oh, well.
Oh,
I think he's left the chat anyways.
Did I forget to send it to Otangelo?
What are you good for,
we
Michael?
need to, if Standing for Truth comes in the chat, we need to make sure that we like get him and say like, hey, what time are we talking tomorrow?
Don't be ambiguous about this. And Michael, I need to know the times.
Time what?
I need to know the times, like for when I'm talking with him about genetics.
Oh,
because I'm gonna need a lot of beer.
Oh, okay.
Well, I know me and Standing were wanting to talk tomorrow. And I have a pretty good PowerPoint going. I'm not going to reveal too much of my hand right now, but
That's
if
nice.
the live stream ends, I could show y'all later.
You guys are
Yeah.
talking about Neanderthals?
Yeah, we're talking about Neanderthals again, and I'm just going to like hammer home the same points I made last time, except now with more data. I asked him if he wanted to talk about like pathologies, reliability of the fossil record, with regards to like human evolution and stuff like that. And he chose to talk about Neanderthals again. So, that's on him. Because like I said, last time after we had the conversation about Neanderthals, I read a shitload of papers. And now I have a bunch of sources that I can pull from. So
We got another one, I think. I think we got another one.
So
another one what?
I'm
Talking
not
let's have a debate on that one, because I'm pretty sure you're full of shit on terms of what I'm actually talking about in terms of the error requirement or possibilities. So
what is that question referring to?
let's have a debate, bro.
Well, I was talking to CRISPR in the chat, so that he's happy. I've discussed many times with CRISPR. I think that'd be a good debate discussion. So that's something we.
Oh, okay. I should have done it earlier. CRISPR asked helicase. Basically, functional helicases can lose like 50% of their amino acids are not conserved.
Mm-hmm.
And John had made the claim that one amino acid error, just like Nephilim free, would ruin the protein's function. And that's also the case for DDX11. And I'm not talking about the pseudogene. I'm talking about the real DDX11. 50% of the amino acids are different or missing in the mouse, and it performs the same function because it has the same 9 motifs that the active domains or whatever. Anyway, let's see. There was something. There was something else. Do you want to hear him talk about body plans? Body plans.
Shit, if he brings up the Cambrian explosion, I'm gonna go off. Anyways.
I mean, in the general sense, the different
Somebody asked him what a body plan is.
Body structures and
the back in chat.
Morphology changes that have happened. Yeah,
Michael.
i.e.,
I know.
Well, when I talk to you, I need you to respond, Michael.
I'll respond to you in private chat.
I'm not psychic.
I'll respond to you in
I was
private chat.
appreciating Maddox's claim about morphology and body plans, because so far it's just empty words. Body plans are the changes in body structure and morphology. It's like, all right, now you're gonna say something?
Oh, wait. Individual proteins have no function alone. Oh, is that so? Oh, please hop onto the stream. I really want to have this conversation.
Did you check the private chat?
This is...
I want to hear what Maddox's answer is to what is a body plan.
Yeah,
Tangelo is not fucking getting in. Really? Come on.
Different body structures and the morphology changes that have happened, i.e., difference between a whale body plan and a lion. I mean, it's rather obvious.
Okay.
What has a lion body plan? Does a cat have a lion body plan? Does a hyena have a lion body plan? What is
Dr.
Does a tiger even have a lion? What?
I'm guessing that Maddox is not going to pass your 10-question test.
Well, that's why Maddox isn't taking the 10-question test. There's no way in hell.
The whale body plan and the lion body plan. I suppose, well, we could give him that, but no, I don't want to.
Well, okay, so generally, like, the term body plan,
more like
it refers to the phylum, right? Right. Body plan's ambiguous, I agree. It doesn't really mean anything. But like I said, normally it refers to the phylum, right? So the phylum is where you find, like, very generalized characteristics that obviously characterize that phylum. So, like, for us, our body plan is segmentation, a notochord, stuff like that. Like a notochord that goes past the anus specifically.
Oh shit! We just got demonetized. You said anus.
It's a biological term, YouTube. Don't worry.
But I don't remember everything.
The medulla,
but like I said,
all this other crap.
Yeah. But it's basically like us and cephalochordates, which are basically like little worms with notochords, are both sharing the same body plan. So according to science, if you consider the phylum a body plan, there's no difference between the whale and the lion body plan. I think the word body plan is very ambiguous, though. I don't think it really means anything.
I just got to put this in chat. Somebody said anus.
Oh, I see Your Inner Fish by Neil Shubin. Love that book. That was the book that convinced me of human evolution.
Yeah.
So, good one.
My favorite quote from Insane Clown Posse. Anyway.
My favorite Insane Clown Posse has got to be like, what is it? Earth, fire, air, and dirt magnets. How the fuck do they work?
Probably one of their most well-known verses.
Yeah.
What mechanisms are responsible for cell fate differentiation? What do you want me to get the book, Otangelo?
Environment. That's one of the factors.
Cell fate
Believe it or not.
differentiation, or just cell differentiation?
Cell differentiation.
Cell
Gradients.
differentiation. Yeah, it's mostly like protein gradients, right?
Laminin, extracellular matrix.
Because the only one that I'm not like biochem person, but the one I'm familiar with,
adhesion molecules,
and of course, like correct me if I'm wrong.
Substrate adhesion molecules. Lots of things are responsible for differentiation. It's a process. Physics has a hell of a lot to do with it. Soft body physics is a very big part of how organisms form.
The one that I'm specifically familiar with, like I said, correct me if I'm wrong, is a Sonic Hedgehog. Like the more, like it's produced in the core, and the more distal you get from the where it's produced, obviously the less concentration, or the lower the concentration is, and that's where you start like fractaling and fragmenting into like arms and legs and fingers and toes and stuff.
Yep.
I mean, it's a very complex interplay of lots of different chemicals. It's obviously chemicals, though. There's no like, it's naturalistic. I'm sure most creationists would agree.
If you want to have fun, get Nephilim to say mesenchymal.
What?
Mesenchymal?
Mesenchymal. MESENCA. I can't fucking spell it right now. I'm too tired. I haven't slept for weeks. Mesenchymal. Mesenchyme. He says mesenchymal, and then he says it a different way every time he says it. It's fun. I love Neph.
Wait, what context did that come up in?
He was talking about embryogenesis, and he was trying to say mesenchymal.
I always love it whenever like they go into the live streams and they don't know how to pronounce the words in their own like slides and stuff.
Yeah,
that always kind of amuses me because you can tell it's just copy and pasted from like Wikipedia.
Let's see, where the hell is Otangelo accretion is? How do you know? How do we know what? How do we know?
That only genetic information is responsible for cell diffraction or cell differentiation.
Well, the mother too. The mother's responsible, and the zygote. Some of the proteins are from the germline, and there are also physics and environment is responsible. So I'm not a DNA done at purist in that manner.
Otangelo asks at Creationist, how do you know Creationist? Because I teach this stuff for a living. That's so good.
Okay. Yep. That's how I make my living. Okay, well, listen, just a little clip here. I'm not sure what this is. What is this? Well, this is about chemical reactions. Now, listen carefully.
Oh, boy.
There is no chemical interaction between the codon and the amino acid. It requires the translation mechanism, which is the ribosome. There is no way around this. This is like there's papers written on this whole conundrum.
The tRNA.
Of like, this is the single biggest question about origin of life is how could this have
I don't know about that. I spent the past couple of days kind of digging through it.
But that's not a chemical, man.
The ribosome?
He didn't think so.
Wait. What does he think a chemical fucking is? What does he think a ribosome is?
Oh, wait, so he's saying ribosome is not a chemical?
It's
not. I don't think. Well, he was also saying that like the gene or your DNA wasn't a chemical, which is like
ABio papers.
Not true.
There didn't seem to be like the attitude that you're presenting here is that it's like, oh
Who
gosh,
else is doubting?
It's just not. That's just not the reality of it. But with regard to what you're actually saying,
what did I do? My cat did that.
Your cat's hitting the play button for you.
Who
Thor.
else?
Bad kitty. Thor.
Who else is doubting that he actually took like any biochemistry or genetics at
Oh,
at
absolutely.
MIT or Stanford?
If he took biochemistry at MIT, there's no way he got a passing grade in it because he doesn't know the fundamentals of biochemistry.
The fact that he thinks like that the ribosome isn't a molecule is kind of precious.
Yeah. Anyway, so
And by kind of precious, I mean extremely
I
precious.
mean, he took biochem, but he doesn't know what fucking ribose is.
Ribose, so that's the thing evolutionists bring up as a defense of their worldview.
Okay, I just want to show this one real quick. Where are we here? Share screen? No. What do I want to share? What's happening? I'm working on this.
Wait, so we have 2 PhDs in the live chat talking to Otangelo, and Otangelo's just like, nah, you don't know what you're talking about.
Otangelo's
like active like, researchers and professors.
Yeah.
Come on, Otangelo,
okay.
humble yourself a little bit. Just try to be try to be not so salty, okay? We know you got humiliated when you came into our first conversation, and it's all right.
Actually, I Creationist Dan says my professor ratings are excellent. I agree with that. I saw them and they were like 4.1 or 4.2. That's pretty good. Really, anything over 4 is like pretty solid.
It's not like, oh, wait a minute. Oh, yes, components and genetics, logic gates, Maddoxism. Why do I have it here?
Maddoxism?
I call them Maddoxisms. Oh, I'm choking. I'm dying. I'm dying, man.
I looked it up. There's 4.3, so even better than I said.
Okay, so I think I got the right. Do I have the right thing shared? I do. Okay, let's listen to this just for a minute. 21. Did I share sound?
Molecularity, Douglas Axe. He was at Cambridge
for
12 years while he was developing, doing this rather massive study on the probability ratio, or the ratio of functional to non-functional proteins, and I believe it ended up coming out to like 10 to the 77th in terms of functional to non-functional.
Hello?
Yep, you're still here.
The speed?
Speed, what's going on here?
Okay, it was 10 to the 72nd to 10 to the 77th, and they always pick the high end of that range, but honest people will say 10 to the 74th. But anyway, Doug Axe did some sort of a study on figuring out what in the protein space, if you started changing the amino acids, basically there'd be a certain amount of functionality. And Douglas Axe did a paper on that, which they quote all the time. And it was, I think it was like, I don't know, 2004 or something like that. Well, Douglas Axe did a lot of really good work before he sold out and started writing books for these guys.
Mm-hmm.
Let me see here.
Oh, shit. The one thing that we haven't talked about was whenever they apparently were like being really sexist towards Jen Aldrich. Do you want to go over that?
Yeah. Oh, God.
We haven't talked about that on live stream. I never saw it.
Yeah, we did. Yeah, we
didn't. We haven't.
I think we covered it very well.
I don't think we did.
Jen Aldrich was on the stream when I was talking about it.
That must have been a like you didn't play clips of what they were saying. You just or you were talking about it in a morning report.
Oh, really?
Yeah.
Oh my goodness.
What's up, Brainbug?
Let's see. Brainbug's here. Okay. Anyway, so you see right here. Now this is a paper from 1996 where they did roughly the same thing with an active barnase variants, and they said, using a sensitive biological screen, we find that a strikingly high proportion of these mutants, 23%, retain the enzymatic activity. So 1/4 of the mutated protein, I mean, they kept mutating it every which way they could. 1/4 of them retained their activity. So that's a lot different than one protein in 10 to the 74th. Also, it was 150, I forget, so it's 20 to the 150th power is how many possible proteins there are. And if 1 in 10 to the, do you know what I'm talking about? If 1 in 10 to the 74th was active, that means a hell of a lot of proteins are active. Something like.
How is there even an upper limit to the amount of possible proteins? Like,
because
it should be infinite, right?
No, you got to pick a size. You got to pick a size for a protein, like 150 amino acids.
Oh,
will give you 20 to the 150th, right?
Well, so, that would be like 150 factorial or something is how you find it, right? Something like that.
20 to the 150.
There's 20 amino acids that can be like conventional amino acids that can be used. Mm-hmm. And for each of those, like you have 150 you can choose from. So it's 20 to the 150. The reason it's typically considered 150 is because sometimes it's named as 100, but like once you start getting very long peptide chains, like 100 or actually even shorter than that, proteins, like especially in the solvent they're in, which is usually water, like begin to fold. Like they begin to try and fold in order to minimize, the charge that's on them.
Mm-hmm.
And it becomes very complicated for it to like try to add more amino acids onto it, unless you have something that's like just keeping the amino acid linear, which is energetically like very costly. Because amino acids, as you well know, they like you have hydrophobic and hydrophilic amino acids,
mm-hmm.
And the hydrophilic amino acids typically want to be towards water, whereas the hydrophobic domains of the amino acid or of the protein will fold in on itself to avoid water. Yeah. And it just becomes a very like spaghettified little mess if you're doing it in solution. And so there becomes a limit to how much you can add before you get a reliable like chain of amino acids.
Yeah. There are a lot of non-synonymous substitutions that can be made to a protein, and in fact big chunks can be cut off and you can end up with the same functions. So
I mean that's the classic thing, like something like 99% of all like point mutations are neutral, and then of that like another 90% are neutral just because they don't substitute the amino acid due to just wobble base pairing or something, or the amino acid that they substitute doesn't matter.
Is somebody peeling their microphone?
Yeah, it's immutable. You could mute him or something if you need to.
What happened?
Mutable? He's immutable.
You're just, huh?
I'm what?
You're immutable.
There was like some scratching sound coming from your mic.
Oh, okay. I don't know what that was.
Peeling your microphone.
Do you want to do it again?
No.
You're good now.
Well,
stop it.
Let's pretend it didn't happen.
Okay. Do you want to watch the Jen Aldrich thing? Or their response to Jimmy Snow and Jen Aldrich?
Yeah, that's gonna be a lengthy discussion.
Okay.
And I'm not prepared with all the chat and all that stuff, but I could bring. Okay. But anyway, let's just do one Maddox.
We can do that some other time. It's chill.
Maddox speaking. So I mean, we can do it tonight. It depends. If anybody's interested in the chat, let us know. If you want to hear.
Well, I was just curious because, like, from what I heard, I assume it's pretty bad.
You haven't heard it?
No, I didn't listen to it.
I haven't heard it at all.
I feel like it would piss me off too much, and I can only stomach like sexist shit once. You know what I mean?
Well, in that case, you're not going to like Maddox. He's just, the way he talks about like women in the role of, I guess, I'm assuming like he holds like very 60s views in terms of like women and their role in the household, and stuff like that, which is just absolutely disgusting.
Yeah, I'm not going to assume. But I would not be surprised. You know what I mean?
No, I wouldn't be surprised at all.
Maddox did not participate in that as much as the rest of them.
Yeah.
Which we have to give him credit for. So. At least I don't think so. I might have missed something, but it was mostly Smokey. It wasn't Maddox. So we should note that. Let's see, Maddoxism. Anyway, this is just Maddox speaking in tongues.
Embryo development is in play.
Whoops. What have I done? What have I done? What's up, Maddoxism 1?
Yes, regulatory components in genetics are recognized as logic gates, and they are recognized as actually Boolean logic gates, and they have
What is a bushy Boolean logic gate?
A bushy Boolean?
What's a bushy Boolean? Is that like a type of cocktail drink I haven't heard of before?
He said.
Yes, I'd like a bushy Boolean.
Bushy Boolean. Ian, is that an Australian thing? Bushy Boolean?
Why would Maddox be referring to Australian like drinks or something?
Sure. Anyway,
then logic built in, and they actually are more complex and have higher degrees of variability and complexity than what we use as logic gates in our current computational technology.
If it's Boolean, how can it be? Is it Boolean just true or false? How can you get more complex than on or off?
Because I believe, and correct me if I'm wrong, Michael, but you can combine like Boolean like operators together to form like more higher order functions, if I'm not mistaken.
Right. And using the idea of logic gates on enzyme pathways or biochemical pathways is a little bit retarded. Yeah, it's a little more complicated than that. There's all kinds of feedback mechanisms. There's analog. There's just a ton more to it. Logic gates are on and off, 0 and 1. This is not compared to binary. I mean, you can find some areas that are kind of binary, like something will shut something off, but no. And if you look at the mTOR pathway sometime, if you want to see something crazy, Ian Chen's never heard of bushy Boolean. I think he just misspoke. Not entirely sure. So Maddoxism number 2 is right here, and I think this is the one that we already called out. Yes, okay. This is the one we have to look at SFT's book. So first of all, we'll play Maddox number 2. Maddoxism number 2.
Okay, so if this temporal variable, i.e. the timeline of embryo development, is in play, then have this regulatory component execute this, express this gene in this manner. When this variable is no longer in play, then move said control variable to a different section of the genome.
Okay, but it's not
reinsert.
Okay,
This is a fundamental issue, and now have an entirely different outcome be executed through that regulatory component.
I'd be willing to bet you.
That's the literal.
What was the safe word?
I don't know. You haven't said the safe word yet.
Still foreskin.
Foreskin.
Foreskin every week. You don't want to confuse me. Anyway, okay. So
have you been selecting foreskin, Michael?
No, but I think John has.
Oh, well, that's okay. Maybe he has. I don't want to like kink shame, but
isn't that where we got it from? I think we got it from something he said. I'm not entirely.
It was because you were messing with him about foreskin, right? You was saying like foresight.
Yeah, foreskin. Okay.
You confused foresight for foreskin.
Kind of like I got short tandem repeats, short condom repeats.
Yeah.
Short repeated condom
repeat. God.
I have been correct.
Really obvious.
I was wrong about that. There is nothing called a short condom repeat in genetics.
I mean, you might hear that thrown around at a bar now and then, but I don't think you'll hear it in a genetics book.
I admit when I'm wrong. It was funny because I really had him going on. But anyway, okay, so he's saying, okay, he's always talking about variables. When the variable is no longer in play, then move.
Oh, that's right. That's what I wanted to say.
Send control variable to a different section of the genome and reinsert with retrotranscriptase. We'll give them that. It's reverse transcriptase, not retro.
Yeah. The thing is, like, most biochemical pathways aren't based upon, well, they can be actually sometimes. most times like they aren't strictly based upon like the presence or like non-presence of a compound. Like the strength of an enzyme usually depends on if there's enough of a molecule to activate it. Like there are certain like kinetic parameters that actually tell you, or like that gauge for you, like how much of a molecule needs to be active for an enzyme to have catalytic activity with it. And obviously, if that concentration isn't high enough, you're not going to have catalytic activity.
Yeah, but here's the important part. Move said control variable to a different section of the genome and reinsert it. Okay.
What? Is he referring to like retrotransposons?
Yeah. Okay. After there's some regulation on or off. Okay, down regulate or up regulate with a variable, as he calls it. Then you move it to another section of the genome and reinsert it through retrotranscriptase. Now you have an entirely different outcome to be executed through that regulatory component. That's the literal of if-then. This is his example. And of course, Gallus engineer is not a geneticist. He's not going to catch that. But do you see how ridiculous that is? Now I want to show you this. Let's see. And that's when Dr. Dan was talking to Dapper Dino, they said that SFT had it in his book. Can you see that? Let's see. I got to get rid of the beef bouillon. His bushy beef bouillon on the floor. Okay, can you guys read that? Another amazing observation regarding genes and transposons, that many retrotransposons, let's see, for many genes in the brain, these jumping genes can jump around the genome, turning on and off genes, performing functional roles. I mean, Dr. Dan.
I mean, sometimes
if I'm wrong,
sometimes transposable elements like paste themselves into.
Well, yeah, that's what they do, but they don't do that in order to regulate a cell.
They don't. Like there's nothing telling, like directing a gene like to paste itself like in the middle of an actual functional gene. Like sometimes it'll do that, but usually it won't. Like it'll just translate, like it'll just paste itself in another region of the genome that's more or less junk.
Yeah, they're suggesting that it's an actual regulatory mechanism of the cell, that the cell does this. Oh, you found some papers on? Did you actually find papers on that?
On foresight of retrotransposons and stuff? Or?
The retrotransposons do not accurately jump to a certain spot.
Well, yeah, I mean, there's no way they could.
So there's no way they could regulate cell function. And both standing...
Oh, they are supposed to. Okay, interesting.
What's that now? Sometimes they mobilize, but it's when the epigenetic suppression fails, then they tend to cause problems. Right. It's going to be problematic. It's not going to be something you can count on for development of an embryo, like John and SFT are both claiming here.
Oh, wait, but they are supposed. Huh? But they are supposed to. They are or aren't. Okay.
You say they aren't.
Yeah, they aren't supposed to. Okay.
Okay, Dan, come in here and teach this to us.
Yeah, explain retrotransposons.
Huh?
Explain to me how viruses are so complicated and why they exist.
Yeah, it's not something that is good. And if it did occur for development, it would keep changing. I mean, it would be a somatic mutation, basically, that would accumulate, and it would be a problem. So,
Brainbug's asking if you could mod Dan so that he can link the papers.
Mod him, Dan. Mod him.
You should mod Brainbug too. Oh, yeah. Standing is here. Standing, can we confirm on a time for tomorrow?
Who the hell is Mod Dan? Where's Dan Mod? Okay, how do I mod Dan? Should I report Dan or mod Dan? Remove, add moderator. Yeah, I think Dan's a mod now.
Viruses exist for equilibrium with bacteria. There could be no life without viruses. While I agree, that's an interesting thing, Otangelo. Who on the Ark was infected with all of the viruses so that they could pass them down throughout history?
Dan, did I give you a wrench successfully?
Can you hear me? Am I here?
Hello. Yeah.
You're here.
All right. Sorry, I'm on a different computer, so I don't have my camera.
Good.
My apologies.
No, you're
got
fine.
cameras on, so.
Okay, you do have a wrench, I believe.
Thank you. I do have a wrench. I can see that. Thank you.
Okay.
It's camera day when Demeter gets here.
So with the what were we talking about? Sorry, hang on.
Retrotransposons?
With the retrotransposons that are supposedly I'm doing air quotes here, but you can't see that. The
It's
functional.
fine. I understand what you're saying, my guy.
What it is, and I'll look for the paper here. I have stuff spread out over too many different devices and desktops and download folders and things. But what I found was that sometimes during development the transposons mobilize, but the vast majority of the time that they do that, they cause a problem. Right. They're not supposed to mobilize, and generally they are inactivated via methylation, via epigenetic regulation. And when that epigenetic regulation fails is when they're able to mobilize. Now, in the book, Standing implies that this is a kind of general component of developmental regulation. It's something that's quote unquote supposed to happen, that it's planned, that it's purposeful. Right. But the research just doesn't support that in any way. It's something that is incidental when it's an antagonistic relationship, right? Transposons are genetic parasites, and the cell that is the host for these parasites, which is to say our genome, is selected to prevent them from propagating. Mm-hmm. And when they propagate, that can cause problems. And that's what's going on here. It's not a thing. They can have effects. They can have phenotypic effects. They can have developmental effects, but it's not a thing that is selected for these things to jump around in an active manner and regulate, for example, gene expression or developmental processes. That's not, quote unquote, their function. It's their function is to replicate themselves, and they incidentally affect the host cell when they do that. But the host cell, our cells, try to prevent them from doing that because that's a problem.
Right. And if they did, it would be random. It wouldn't be controllable. There's no way an organism could count on developing the same way twice, right?
Yeah, the insertion sites, there tend to be hot spots and cold spots, but they tend not to be sequence specific.
They're a little fuzzy. Yeah.
Yeah.
So you never count on that. So it's just a ridiculous statement. So that's why it is a Maddoxism.
Yeah, it's I'm going to mute myself, and I'm going to take a few minutes to try to find the specific paper here because I found this exact thing, and let me find it, and if I can find it, I'll read you the relevant quotes. Give me 1 minute here.
Okay.
Okay.
Here what is Standing? Here what is?
Oh, Standing doesn't have a wrench, so he can't post the
Oh, okay. Let me give Standing a wrench.
No.
Oh, well, is he going to send porn now?
Yeah. He might try to porn bomb us.
He's going to porn bomb us. You got a wrench,
Porn bomb us with creationist material.
You got a wrench. Did you get the link speed? No. Standing, I have so many windows open right now. I have no idea where my mail is.
Post the link to the article.
You have a wrench.
Yeah.
I'll go back to that window. Where is it? Let's see.
You get a mod. You get a mod. You all get mods.
You all get mods.
We'll give anybody a wrench. Let's see.
Anybody except Otangelo. No.
Oh.
So Otangela's going to have to work for his wrench.
Yep, he's going to have to work hard for a wrench.
No gish galloping, Otangelo. That'll be step 1.
The paper he sent's not behind a paywall. I mean, I'm not.
Well, you know how to get behind paywalls.
Well, if you
the link he sent's not behind a paywall. It might just be because I'm logged into my school and it just picked it up on that database. But I don't know.
Oh, let's see here.
Oh, actually, yeah, that's exactly what happened. I have full access to the article because of my school.
Let me see here. Do I have
I'm
that?
surprised. That's the first time it's ever actually just worked from a link. Usually I have to like log in, and yeah, it's good for me at least.
Is your church also on campus?
My
Let me
church.
see if I.
Wherever you are.
Hey,
Oh,
So
I'm not
this
on
is
campus.
this
right
is
now.
the exact paper I was looking for. Can I highlight the relevant paragraph in this paper for y'all?
Yeah.
All right, so you go down to page 4, okay?
Hold on. Let's see.
Wait,
Yeah, you
screen.
kind of have to. It's a couple clicks, but you can get to a PDF here.
You're not sharing Dan's screen or anything.
No, I'm not. I haven't shared anything. If I
Oh,
can
okay.
actually, I can share it. Actually, give me 1 second. Give me 1 second here, and I will share it.
PDF is not behind a paywall. You can
Boom.
download it, the one that Danny sent.
It's this one.
Okay.
Here you go. All right, here you go.
Let me go over there.
He's sharing his link.
I have found the paper. Hold on.
Did you share it in private?
I screen shared. If you want to pull my screen up.
Oh, okay.
Ah, here we go.
All right. So here is the relevant paragraph. If you go right here, this paragraph right here, our results provide clear evidence. So somatic L1 and ALU mobilization. Okay, great. So they mobilize somatically, which is to say it's not in the germline, it's during an individual's lifetime. Fine. But I want to highlight this sentence right here, because this is the key down here. In contrast to germ...
Oh, sorry. I was
Say
going to
that
ask
again?
if you could. I was just going to ask if you could read it because I can't really see it very
Yeah,
well.
yeah,
But
I'm
you're
gonna read
reading it anyways.
Yeah. In contrast to germline activity, somatic insertions disproportionately impacted protein coding loci. Now, creationists, I think, will be the first to tell anyone that mutations in protein coding genes tend to be harmful. Germline insertions are rarely found in regions where they generate a deleterious phenotype because such mutations are strongly selected against during evolution. That makes sense, which means the instances of these entities in our genome, they tend to be in places where they don't matter. Because if they were in a gene or affecting a gene's expression, then it tends to matter. And that could be harmful, and then it's going to be selected against. So somatic events, on the other hand, and the on the other hand is key there, on the other hand, are present for one generation and may affect protein coding loci in a specific context, perhaps drawn to open chromatin or transcribed regions. Now, I'll get to the second half of that sentence that I've highlighted here in a second, but the first half of this sentence is the important part. Somatic events, on the other hand. In other words, germline ones tend to not do anything. They tend to not break anything. Somatic events, on the other hand, meaning somatic events tend to cause problems, meaning they're not selected for to do like run-of-the-mill day-to-day regulation. They tend to be accidental and they tend to cause problems, and then are present for one generation, as in they are selected against rather than propagating, and they're drawn to open chromatin in transcribed regions because the enzymes that are going to pluck something out and put it in, where they're going to put it in, it's not going to be the densely packed heterochromatin that's not doing anything. It's going to be the open to other enzymes, euchromatin, the genes that are being expressed. So what this paragraph explains very clearly, if you know how to parse the language, is that the ones that are persistent in the genome, the ones that everybody inherits in the germline, they don't do anything because they're in parts of the genome that don't affect anything. The ones that happen somatically tend to cause problems because the targets where they can insert tend to be the regions that are active, which is to say the regions that are functional. And when they insert, they tend to break those regions, and then they don't persist because those insertions are harmful, which shows that, contra what Standing for Truth is saying, this somatic mobilization of these elements is generally harmful rather than part of the run-of-the-mill gene regulation that we find in organisms.
Okay, so what is the, could you link this paper or show us the name?
I'll stick it in the chat here.
Could you scroll up so we can see the name and the...
Yeah, here you go.
Okay. Somatic retrotransposition. Oh, that's the same one that Standing just sent to me.
Yep, that's the one. Yep. Yeah, no, it's Standing's own paper that he cites saying one thing actually says the opposite thing.
Okay. So, midnight, we will send you the papers. It's a free
I just sent it to him.
Huh? Oh, you sent it to him? I made
Yeah,
it.
it turns out the actual paper itself, like where it's published, it turned out to be 4 papers long. But I think if you access it through PubMed or something like that,
Yep.
it turns out to be like 11 or 12.
Yeah.
That has an interesting problem at times, like getting.
Anyway, it just doesn't make any sense to say what Matt
Dan left.
said. And Dan's gone. He's still.
No!
He had to go. He's back. He's back.
He's back. Thank you.
I was thinking it was like a "I must go, my people need me" thing.
No, that may very well happen soon. I have the baby monitor on. But so I may have to run. But yeah.
So did you send a different paper, or did you send the same exact?
I sent the PDF of the same paper.
Oh, okay.
Okay.
Steve, Standing has a question for Dan.
I am not familiar with Carter's analysis of that paper, but I just parsed it in some detail for everybody, and I'm confident that my analysis is correct. So let me put it that way.
I'm confident that Carter had a hidden agenda when he parsed it. So
Is Rob Carter a creationist, dude?
Rob Carter is a young Earth creationist geneticist, I believe. He co-authored a paper with John Sanford on H1N1. That is my favorite incorrect paper of all time. There's literally nothing correct about that paper, and I love every word of it.
Yeah, you gotta quit reading those guys. You gotta branch out a little bit and get some other opinions from the other side of the fence.
Little shameless self-promotion. I have a video on that paper. I cannot describe enough how wrong that paper is. It is incredible.
Is your a video on your site?
It's on my channel. It's one of my creation myths is H1N1 extinction something or other.
It was one of the first ones, right?
It has to. Yeah, it was pretty early. It has to do with genetic entropy, and it's that there's nothing correct about that paper. It's a monument to wrong. It's amazing.
You do realize I link to your site all of the time.
Yeah,
I appreciate that.
Okay.
Yeah, my only I don't remember it super well, but I remember one of my takeaways is they measured fitness with virulency, which is just such a weird metric.
Yes, that is
natural selection actively works against virulence.
Yes, that is a terrible way to measure viral fitness. It's so bad.
Yeah. Oh God, it's exhausting keeping up with these guys.
Is it really, Michael?
Carter implied liver cell polyploidy.
Yeah, what the hell?
Interesting.
Due to retrotransposition, which is false.
Yep.
Let's see here. So we have Sexy Killzone in the chat. What else are we doing, guys? Let's see. There's other stuff I want to talk to you about, Dan. I can't remember. I sent you emails. I have no idea.
I apologize. So for people that don't know, and if I've been unresponsive to things, my wife and I, we had a baby about 2.5 weeks ago.
Yeah, that's right. I forgot about
baby. Wow.
Thank you. So we've been busy and sleep deprived.
Oh yeah.
So it's I'll pop into random chats at like 3 a.m. when I'm feeding the baby or something, but then I'll be like asleep in the middle of the afternoon when people are sending emails. So it's been a little hit or miss lately.
Can I just say that you're very polite?
Yeah, I appreciate that.
It's not a bad thing, obviously. It's just
I don't know how you keep your cool. And in some of the hangouts I've seen you in, me and Walker were screaming like.
Yeah, that's my thing. I think I'm generally like a nice person, but I don't know how long that would last against smoky aromatics.
Yeah.
Like I just don't know how I'd be able to keep my cool.
There must be something about like the PhD process that like incredibly like humbles and teaches a person like immense patience.
That is definitely the case.
Yeah, because I know like I've talked to some of our professors like when I was an undergrad at my university, and they always kind of give that same kind of calmness about them. It's like it's the other side of it is the normal class I teach is, and at this point it's no secret who I am or what I do or anything, but I teach the general biology program. I'm one of 3 instructors at Rutgers University that does the intro bio sequence. So between me and my colleague that kind of catch all the freshmen every fall, between the two of us, we have between 1,800 and 2,000 students a semester. So I get up like 900-ish students a semester. Ooh, and the questions I get and the conversations I have are just incredible and fascinating and wonderful, and also occasionally frustrating and like puzzling. And there's nothing about the conversations I've had
since I've kind of jumped into the YouTube creationist sphere that are in any way more frustrating and puzzling than the conversations I've had in the context of my job.
Really? That actually kind of surprises me.
And I say that as somebody who I one of the things I've been so has been so frustrating over the last 6 months is that halfway through the spring semester we went remote and I lost the ability to directly interact with my students, and that it made me so sad because even the weird, crazy, out of left field questions, like, they can lead to really good discussions, and it's I miss it so much, and I can't wait till we can just go back to doing things for real.
Yeah,
on the student side, I have the same.
Interestingly, when I was an undergrad, luckily it never happened during any of like the intro bio courses I took. But there were issues at times where where I am, like, it's not too uncommon to have a significant like creationist population that might be present in the classroom, and so you can imagine how well that goes when we start talking about evolution and biodiversity. And according to the professors there have been a few instances where they've had to actually remove a few students from their class because.
Interesting.
Yeah,
Are
because
they just being like rowdy and stuff and not letting them teach.
Not even just rowdy, just like interrupting, like whenever they get the chance.
Like heckling?
Oh yeah.
Wow, that I've not had to deal with. I have had my favorite moment so far was one day. There was a student on, I guess it was like a Switch or something, like a Nintendo Switch, and they were playing. I think it was Mario Kart, but they had the volume on, like, in class, and you could picture a lecture hall that seats 500 people, and they're in there playing Mario Kart, and they hit like a banana, and it made that like wow 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 like banana spin out noise. But the volume was really high, and the whole room heard it.
Oh man!
So I had to be like, look, don't play games in here, please. So that was probably the most in terms of like that kind of thing. I don't get students that are hostile. That's never happened.
Yeah, it's very rare for it to have happened, but there have been instances of it. Like that people like that I knew as TAs like have kind of like told me, it's like, how's your job as a TA? Well, today was kind of interesting.
So if I can, sorry, I don't mean to draw this completely off topic, but what do you TA? Because when I was in grad school, I was a TA for all 6 years.
I'm not a TA, but I knew several people in the chem department who did TA.
Gotcha.
Hopefully, if I'm enrolled in a grad program, I'll become a TA and have
it's very much worth doing, but it's a trip.
So
I can imagine.
I have a question for you. In your conversation with Smokey yesterday, you mentioned a biology stack or something. Free.
I'll pull it up. It's OpenStax Biology here. Hang on. I'll pull it up and put it in the chat. It's a really nice open source textbook.
Yep. I was trying to find it, and I couldn't.
I got it right here. I'm putting it in the chat. I got it right here.
I also do have a general question that, like, I'm asking it for molecular alchemy because he was wondering about this. But what does like your school think about your YouTube channel, if they know about it and stuff? And
so
what is the response?
Well,
there?
So the Creation Myths channel nobody knows about because that is a post-quarantine thing.
Okay.
So like nobody knows about that. I will say I do have another YouTube channel, which is paintball oriented, and paintball.
Paintball. Okay. Nice.
And at the very end of last semester, my students found it, and that was one of the funniest things I've ever like. It was hilarious in their on the subreddit. Someone made a thread about it. Really? In like the group chat for the class, they were like, "We found Dr. Stern Cardinal's YouTube channel," and then they started making memes out of screenshots from it. It was. Yeah, it was a whole thing. It was really funny.
At my we found my Calculus 2 professor's SoundCloud.
Nice.
But I was wondering, like, does your school approve of participating in conversations like directly with creationists and that kind of stuff? That's what molecular alchemy was wondering.
Yeah, that's, I honestly have no idea. And I don't know that, I think it's very much a person by person kind of thing. My department is really good. I love all the people I work with and my supervisors and everything, which is not something a lot of people in academia can say. I'm really fortunate to work with a bunch of really, really, really great people. And my impression is that they kind of, as long as you're on top of things and doing right by the class and the program and the students, then, as long as the extracurriculars are not a problem for the school in any way, then, whatever. Yeah, I don't know beyond that. Because again, I literally have not seen anybody in person in 6 months. So we'll see maybe when we get back in I don't know, January or something.
So y'all aren't going back in the fall?
Right now, everything is remote. I think through the fall, and then who knows? Fingers crossed. I don't want to be remote again in January. I've had enough of this.
Oh man,
we're supposed to go back, but we'll see how long it stays. What were we saying?
University of Minnesota was going to go back in the fall, but then I guess some frat houses started having contests about to see who could get COVID.
Oh.
They canceled.
Oh Lord,
yeah, that happened
in Florida too.
Yeah, I'm in Alabama, like the pinnacle of SEC culture. And like Auburn, Alabama, UAB, like they've only been at school for a week and they're probably already about to get sent home.
It's a bunch of schools now that have opened, they've had an outbreak, and after like 2 weeks they send everybody home.
Yeah. The university I went to recently had a professor who essentially said like COVID isn't a real thing. Something. The academic equivalent of saying that was as professors will say, skeptical of it. Yeah, we had one of those. That was
oh my god.
Yeah, and recently we found out that the football team apparently has some members that were that they knew had COVID and are still in the football team. Yeah. Florida is quite the state.
My school's still planning on going back, and we're they have to milk us for money in some way. But I don't know. We'll see how long it stays, because my school's generally more on like the liberal side, so I feel like it would be pretty quick to shut down compared to like SEC. But it's also a lot smaller, and generally the students aren't, at least I would hope the students aren't stupid enough to go and just have like massive frat parties and stuff. I mean, there's no way they'd be able to. I feel like the school's too small and like regulation would be too tight,
Yeah. So Dr. Dan mentioned like you have like 900 students. That's interesting. Like the university I went to, I mean, like the max we held was like 330 students at a time.
Yeah. Well, I think he teaches like multiple classes.
Yeah, it's multiple sections. Yeah. So every year, every fall and spring, I have 2 sections of this general bio, Gen Bio 1, Gen Bio 2, and each one in the fall for Gen Bio 1, the enrollment is usually pretty full. So it's, in theory, if everyone comes to class, it's going to be like 450 per section. And then in the spring, there's a bit of attrition. So you're still looking at like 350 to 400.
Is that all the pre-meds that are no longer pre-med?
It's everybody. It's pre-med, it's biomedical engineering, pharmacy, any life science, which is everything from like cell bio development, genetics,
no, I was just saying,
biochem.
Like you said that there's like a pretty large drop off between first and second semester, and I'm saying
the other thing,
kids who were
it's in order to go to part 2, you need to get at least a C in part 1.
Oh, yeah. Okay. Yeah, I just know
that's a common thing.
Yeah.
So I'm trying to find the most in-depth thing I can on recombination and linkage equilibrium.
Ooh.
Nice. Linkage disequilibrium?
Yeah, disequilibrium. And I'm very, very tired. But recombination, I'm trying to find something like review papers or something very, very detailed, or something more than what you get in Lewin's Genes 12 or whatever. So if that's something you think about, like that is kind of a private chat that we all like to have with you and Molecular Alchemy, and if you ever get time for us to get together, and do a little bit of kicking around and sharing resources on stuff. But anyway,
yeah, I remember one of the most fun things we did, like with biochemistry, is actually advanced biochem. We did look into a little bit of like epigenetics and stuff. That was fascinating. Such a broad term, but there's so much there too.
I have 2 really good textbooks on epigenetics. What one is kind of thin, another one's really fat, and it's a lot of papers, but they're expensive. That one is, hang on a second, you got it right here. Ellen, oh my God, it's Ellen Caparos, Genuine, whole bunch of people. 2nd edition, and I think it's from like 2018, 2019, something like that. And the thin one is actually probably faster to read, but
what's the title? What's the title of that book?
Epigenetics 2nd edition.
Yeah.
So it's pretty good. The other thing I wanted to ask you, Dan, is the thing with Smokey yesterday, how did that? It came up because he was having a problem with the test, right? But did you notice the that he kept trying to dip you into the naturalism philosophy debate?
Yeah, so we were going back and forth via email, and then he said, "Can we just talk about this, quote unquote, face to face?" I said, "Yeah, sure. I mean, my schedule is a little bit weird, but if you can hop on in like an hour, let's do it." But yeah, then in the thing, it was like we want to talk about
the test,
philosophy of science and naturalism and assumptions and this thing, and it's like, no, I like trying to keep it focused on like the actual challenge. And I will say, by the way, for anyone listening, for anyone who is considering stepping up and taking the whatever we're calling this, the creation challenge or whatever. So our friend / acquaintance, Corporal Anon, who, for those of you that know him, is in geology, not biology. We went through it last night, and he got like 7 out of 10. Like, he did well. So these are not like,
so you do it privately?
Yeah, we did it on just a Zoom thing, and he got like 7 out of 10.
So
it was fine.
How do I sign up?
What type of questions are you asking? If I can ask.
So the ones that so I'm keeping the exact questions, obviously secret, because
yeah.
I want, if the point is to see, like, do you know this stuff?
Right.
But like, I mean, I've said the first question, which is in the original challenge. I said the first question, which is explain how natural selection works. Just like explain how that works. And then I have stated that another one of the questions is to interpret a Punnett square. I'll show a Punnett square and just like tell me what's happening.
Okay, yeah. So fairly basic.
Like, we're not talking about like stuff that you're gonna have to wait to grad school to be exposed to. I'm talking about things that could be on an exam in either like a general biology class or like an intro level like evolution class. Right. It's not super intense stuff. And the point is, in conversations with people, like earlier in this chat, we mentioned the Maddox like thinking that ribose was a protein kind of thing. It's like, if you're going to, like say that you can refute the theory of evolution, like I would presume that you would know the basics of what is evolutionary biology. And my impression is that for a number of people, that is not the case.
Have you gotten any takers besides just Smokey and Nicholas at this point?
Standing has indicated his interest, but also that he is too busy for the next few weeks. Although after he said that, he did like a 9-hour stream yesterday. So,
oh wow.
But yeah, I think so. Standing is busy with stuff, and that's cool. So I'm happy to wait until he's free.
He was sleeping for 4 hours.
But the funny thing is with Smokey and Nicholas, I'm very happy. And if either of you are listening, thank you again. I'm happy that they're willing to participate. But like, people that are upfront about not being experts in the field are not the people I'm targeting, right? Yeah. Because the point is people claiming to be experts when they're not.
That was the thing I was talking about earlier.
To like be able to refute specific arguments when they don't know the foundations of those arguments. And people that are upfront, and I think I've said elsewhere that I disagree with Smokey on a lot of things. I have a lot of problems with stuff that Smokey said, but to Smokey's credit, to be fair, he is very clear that he is not an expert in evolutionary biology or genetics or any of that stuff.
Hey, you forgot the first rule of the Saturday night. No being fair to Smokey.
Right.
Well, man,
no apologies.
Clown if you're fair to Smokey. So
I kind of want to build.
I had really good conversations with Smokey, and then all of a sudden things just went south, and I still am not entirely sure why. And it was really odd, but it had to do with naturalism, and it's kind of odd. Like, what do they want from us? They want us to quit looking for like abiogenesis. They want us to quit doing the research. They want us to do what? Like, we never sit around and say that we're disproving God. I mean, what do they want?
That's the thing that kills me too. I mean, like, I understand like Smokey has his limitations with science. I mean, so do I. I mean, I understand biochemistry to a fair extent, but I don't understand things like isochron dating or like nuclide decay or stuff like that. Like, there's limits to the things I understand. And I don't think I would. But
at the same time, you don't try to debate them.
Precisely. That's the thing. Like, I'm well aware of like my limits in terms of what I can discuss rationally, and it's one of the things where it'd be foolish for me to like try to engage intellectually on those topics as if though I had like an adequate understanding of them.
Yeah. Well, I think that's just generally something that studying science beats into you. You know what I mean? Like.
Well, yeah, I mean, I just don't understand why Smokey thinks, like, especially with this whole abio thing, that he understands, like, the topic better than other, like, that he understands things better than other people do.
Well, there's this perception of a threat from abiogenics research and evolution. And the thing is, what would they have us do but to figure out how things may have happened? And to go back as far as we can, it's confusing about what they would have us do. They would have us quit using some words, and I'm not sure what it is. And it was leaking out in that conversation with you when he was talking about you showing a slide to the students and claiming that was the best theory or whatever.
Yeah, and going about consensus.
It's sort of an odd thing.
I mean, that, it's funny because showing that slide was like, it wasn't even like, here's the answer. It was just, here's an example of an option. And when I do that part in class, it's very much like, here's one of the hurdles that needs to be overcome in order to solve this problem. We don't know what the answer is, but here's a couple of the options. And I have a couple slides of like, here's one option, here's not, whatever. I'm very much not presenting it as this is the answer. Because for some of those questions, we don't have the answer, and that's okay. That's the point. If we had the answer, science would be wasting its time, right? Mm-hmm. Science concerns itself with things where there is not yet an answer. That's the point.
Yeah, and here's the other thing too, like especially with abio. I mean, what I've researched into it, and it's not overly much. I mean, I don't proclaim to be an expert or even well read in the field, but like from the stuff I've looked into, like it seems that this idea of like there has to be one way like to achieve proteins or things like that doesn't even seem to be correct. Like there seems to be multiple ways like that you can get the same thing accomplished, although they all like these chemical processes, like some of the processes or methods used can only foster in specific environments. Mm-hmm. And the question becomes like, okay, are these environments like reflective of what the early Earth would have been like? Or, for instance, okay, if we're dealing with this particular, like hydrothermal or alkaline vent hypothesis, sure, you might be able to get proteins or things like that to form, but can you have them like also encapsulated within lipid membranes? Would those, would certain structures like be able to stably like merge and, form hybrid complexes and things like that?
But then the interesting thing about that question is that there's a whole other school of thought that puts the order in the opposite direction.
Oh yeah.
Right. So there's the structure first school of thought, and then there's the metabolism first school of thought. And so this whole discourse over like, How do you get the membrane? How do you get the RNA in the membrane? How do you get the proteins with the RNA in the membrane? There's a whole other approach to the question where none of that stuff matters. And all that matters is how the things interact.
Yeah, in fairness, like I've heard like arguments like all around the field, and I kind of sympathize more like with the metabolism first people, and maybe it's because biochemistry.
I agree. I will say I 100% agree.
Yeah, because my contention with it is that
Okay.
like hold on.
We have a party
Okay, go
going on
ahead.
tonight.
Oh, by
Yeah.
the way, Dr. Dan, meet Molecular Alchemy. He just popped in.
Hey.
Yeah. So
Yeah,
my
that's not me.
yeah, so, like my contention with other fields or with other like ideology surrounding abiogenesis is the fact that what do you learn in Bio 1? Well, you learn that most processes that occur in cells require energy in some form or another. There are reactions that are autocatalytic, so they don't require tons of energy to get them started or any energy at all. But a lot of the like reactions and processes do require some kind of energy storing molecule to be broken apart. Usually it's ATP. And it seems that if you don't have like some kind of energy bearing molecule, be it like inorganic or otherwise, that there just seems to be fundamental limits to what a protocell could do if it's not able to like use and synthesize its own energy source. And so for me, it kind of becomes, okay, shouldn't the energy source be the thing like that's most contested in this case? Because sure, genome evolution and like how it forms, like how the connection between proteins and genetics becomes stable is an interesting question, of course. But the energy needed for that to occur has to already exist. It can't just magically pop into existence. Like
Right. You need an energy source to facilitate the reactions prior to any specific like cellular, air quotes, cellular or protocellular or whatever structures. The reactions have to be able to happen.
Right.
And I actually had a conversation yesterday with somebody on Discord about this and shared a paper. There's a whole series in the literature about coupling biological reactions to the conversion of sunlight into heat. And it basically refutes the argument that abiogenesis violates the second law of thermodynamics, where in fact it seems to be the opposite, that the production or decrease of entropy is actually coupled to formation of long nucleic acid polymers in particular, in the sense that DNA and RNA polymers are the best chemical structures that we're aware of at converting sunlight into heat. So that maybe
Was
all
that
along, maybe this is not an initiating event, but something that kind of drives the process as it goes, supporting, the formation of kind of the precursors of protocells.
Was that the paper from a few years ago that argued that, or was that related to the paper that argued basically that life is inevitable as a thermodynamic kind of imbalanced discharging mechanism? Was that kind of the gist of that? I don't know if anyone remembers that paper from a few years back. It made a little bit of a splash for a couple of weeks.
Yeah, that's kind of in this niche of the field. The paper I shared, and I can share it with you guys too. I'm on my phone now. It'd be a little difficult, but in the future, is a review kind of all the competing theories.
If you have a name of it, I can look it up.
The one I'm thinking of in particular is titled "The Thermodynamic Origins of Life," I believe, and I think it's 2010. But there have been more recent publications than that. I mean, that's a decade ago now.
Hey everybody, I'm going to take this moment to say goodnight. Thank you for having me on. Delightful chatting with all of you. I will talk to you all soon.
Okay, thanks, Dan.
Well, take care.
Take care.
Okay. Hello.
Hello, Midnight.
Hey, what's going on?
By the way, you mentioned epigenetics second edition. Is that a textbook?
It's textbook, yeah.
Can I just say thermodynamic dissipation theory for the origin of life? Is that the one you're thinking of?
That was an
Yeah,
interesting thing to read. If it's the same one I'm thinking of,
Okay.
I forget the
I'll
guy's
post that in
name,
the
but
chat then.
it was very math heavy.
Is that a paper?
Yes.
Yeah.
Could somebody put the name somewhere or link? We hate to talk about papers unless we can link them.
I put a link in the public chat. That might be the one they were talking about. Pretty sure.
Yes. Yeah. This is the one. This is the one I read. Or tried to read anyway.
I gotta click on it. Let's see where just to walk and fish. Okay.
Okay, $160 on Amazon. Jesus Christ, textbooks are expensive.
Yeah, that's what I paid for it. But you know,
I mean, fuck. If you can give it to me for free, I'll take it.
there are other options.
Well, yeah.
Did you find a PDF of that book, Michael?
I have not looked. I would have to look. Hang on a second.
Yep.
Yeah, Molecular Alchemy, I'm kind of curious on your opinion about this, like between the whole, what is it, like metabolism first, information first, and I think replication first is the most generic title it's given. Like, where do you like fall in between those three camps?
So, and I've taken maybe not a deep dive, but this has kind of been a pet of mine, trying to really survey the literature on abiogenesis and come up with an opinion like you're asking. It seems extremely plausible that metabolic pathways. So let's set the stage a little bit. So what we're talking about is a sterile environment, but it's also an environment with huge amounts of geothermal activity, huge amounts of weather activity. So you have the entire surface and probably subterranean as well, not to mention aquatic environments potentially interacting. And on top of that, I think there's good evidence now of extraterrestrial contribution, so that repeated asteroid strikes can actually create stockpiles almost of different biologically relevant compounds. But in my reading, it seems the most plausible is that you have some basic metabolic environment as kind of the initial step. That if we were back in time and actually observing it, that would perhaps be what we see first. But then the nature of RNA by randomly incorporating different nucleotides to have different activities, even with RNA polymers being as short as 5 to 7 nucleotides, a minimum of that, you can actually have ribozyme activity. And this is not necessarily the self-replicating ribozyme. This is probably catalyzing some simple chemical reactions. But combining a production of RNA polymers with the presence of lipids in an aqueous solution, like right there, those two factors, which those aren't easy factors to meet, but once you have that, it's not hard to imagine the formation of these maybe proto cells, where you have a very heterogeneous population of cell-like bodies spontaneously forming in water and randomly capturing different short strands of RNA, and this happening millions of times a day, or maybe even millions of times a minute.
I think there's an interesting thing.
The right combination.
Yeah, there's an interesting argument that's been put forth, is that So like if you take like a strand of E. coli and you put it on a template of just glucose and you have it replicate, two of the byproducts of metabolism of glucose are glycerol and acetate. And it turns out that the conventional it turns out as you have it replicate for a few days, what you'll develop is that the E. coli tends to segregate into 3 different strands. So one of them are glycerol specialists in terms of their metabolism. One of them are acetate specialists, and some of them are glucose specialists. So some of them are better at manipulating various metabolites than others. And I'm thinking that might be like how chemical fixation, or at least kind of taking care of like this heterogeneity problem, might have started out. Like it could be that you had some cells that had the ability like to fixate certain molecules, and like over brief periods of time, or maybe extended periods of time, you could have it be the case that these protocells were able to kind of fixate the environment to their own liking and kind of limit the diversity of available molecules, so that these these permeable protocells aren't just ingesting anything that's too small to permeate the cell membrane. I don't know.
You know, am I taking up space? Should I leave?
No, you're good.
Midnight, did you see the second book I sent you? The second name?
Wait, no. Let me go check my email.
No, it's in chat. I put it in chat. So. It's Epigenetics by Lyle Armstrong. Check that one out.
Okay.
And I will email you about.
Google is your friend.
Google is your friend.
Epigenetics 1.
It's a little more to the point. It's actually a lot denser. The other one is like a lot of papers.
Okay, I hate e-textbook. Paperback is like $40. Jesus Christ, these books are expensive.
Yeah, that's why I'm broke, guys.
Yeah,
that's why you gotta send me money. Send me money.
Okay, you know what? I'm adding it to the list, and find a not PDF of this not book.
What were you saying, Molecular Alchemy?
Well, I'm just gonna wait for them to get their book
We're done.
squared away.
I think they're good.
We're done.
I think we're good.
All right, no, I think that's reasonable, and I think, you know, things like endosymbiosis. I think we have hints, in addition to all the experimental data, which is enormous, that's accumulated like over the last 20 years, which we should absolutely, with our paper trails discussion, we should really talk about some of these paradigms, some of these discoveries and advancements, because with abiogenesis, I've actually like in the last week, I've heard people from other groups essentially saying that Miller-Urey, they were construing abiogenesis research as though Miller-Urey was the culmination and nothing's happened since then.
Yeah, and
basically, like everything's happened since then.
Maddox or Standing just said today, I think, that in the last 5 years, nothing has happened in abiogenesis.
Oh, and that's so that.
Yeah, that they haven't proceeded. Yeah. And Otangelo said that there are like 8 amino acids that they can't even create, or something.
What? I mean, I believe Smokey asked me about it. I think it was about glutamine. I think, and I was like,
yeah, glutamine was like the simplest one.
Well, he asked me because I mean, it's true that like glutamine, at least from my knowledge, like doesn't appear. Like isn't found in meteorites, but what is known is that glutamine can be formed from glutamic acid by means of transamination.
Yeah.
It turns out that glutamic acid will exchange its carboxyl group for an amine group, and it's like, okay, I don't see like why it's an issue like for one of these molecules to be absent if you have a basic chemical reaction that can convert one to the other.
Yeah.
And we can have really exotic environments, really exotic conditions. That's often one of the critiques of abiogenesis research is, the early Earth conditions. We've got a pretty good idea of the early Earth conditions. What we have is a good idea of average early Earth conditions like 4 billion years ago. That's not necessarily what the local environment of a deep thermal vent would look like. Or there's another paper I can share with you guys that talks about these asteroids and in particular looking at ice granules, these nurseries that can exist on these asteroids. So in the vacuum of space, which, in a synthetic chemist lab doing reactions in vacuo,
I have a quick.
At that pressure, you can get different products from reactions. So their conditions are not
I have a quick.
At the earlier.
Midnight.
Yeah, I had a quick question. Do they know the early conditions for the Garden of Eden?
No, but that's irrelevant because
I think they were. I think they were kind of like I don't know if you imagine that dude laying under the palm tree with somebody feeding him grapes.
And there's a T-Rex crunching watermelons right next to him.
Yeah, there we go. T-Rexes were definitely herbivores, by the way.
They were fructivorous like raw mat.
Wait, yeah, okay. I wouldn't know why in the fuck did the T-Rex have those tiny arms.
I can tell you what has not been established, regardless of how many times Ottangelo may say it. It has not been established that it is impossible for the simplest forms of life to occur naturally by wholly physical and chemical means with no agency involved. That is almost a mantra by some of these people. They start throwing out stats, saying effectively. I know in particular the ribosome is one thing they point at, and somebody may have calculated what are the chances randomly of 50 different proteins and multiple subunits of RNA coming together randomly. But that's a problem setting up the scenario like that, because the evidence we have indicates that form of ribosome was a later step. That what we're really talking about is more akin to viral organisms or pseudo-organisms, depending on who you ask. That you have some of the criteria met initially of life, and then selective forces, because there's variation and replication, selective forces coming into play.
Mm-hmm.
Well, I think the important thing to remember is that like the stuff that cells use now, I mean, like the modern version of these proteins are undoubtedly, I mean, nobody will deny it, they are extremely complex. Like they are very convoluted and the way they're depicted in textbooks look very pretty. I think if you look at the protein structures, they look like somebody threw spaghetti on a wall and called it art. But in seriousness,
I mean, in some circles, that would be considered art.
Yeah, in some sense, it is very beautiful. I mean, the activity these proteins have are very interesting. But we should be thinking about kind of what's the bare bones like type of reaction or activity that some protein has. Like, what's the absolute minimal thing like that this protein needs to do to take chemical A and process it to become chemical B or something like that. I think kind of that's kind of one of the points like that can't be stressed enough is that if you have that basic process, like if you have this underlying like simple process or chemical process where A can be transformed into B, once you have that, you're golden. Other things can develop. Other things can attach, around the surface and play regulatory roles or be part of glycosylation or other things. But those things aren't a requirement for the catalytic activity that, takes A and spits out B.
So we have somebody in chat who actually wrote papers on abiogenesis. Demeter, 11th dimension. He said we need to set up a QA chat only for abiogenesis. I think the people do not ask the correct questions.
No, I mean, what's curious, molecular alchemy might be able to remind me if this is right or not, but it doesn't seem like that even Maddox, when he was talking about proteins and amino acids, seemed to understand that proteins, or that amino acids come in two, alpha amino acids come in two different flavors, like L amino acids and D amino acids.
Mm-hmm.
Which,
well, what do you mean?
Yeah, he talks about the chirality problem all the time, but that is
which may not actually be a problem. I mean, there's I'm not aware of any evidence suggesting that it couldn't have occurred the other direction.
Right.
Once you have a self-replicating population that just happens to be based on left-handed versus right-handed, you can see how something like that can persist.
Mm-hmm.
I thought they were less stable, the D's, for some reason, and I'm not sure. I read that somewhere in one of my protein books, that it was less stable, and that it would be selected out, at some point. So,
well, if that's the case, then that may be it right there. But speaking of 11th dimension, this comment he said about people not asking the right correct questions. That's absolutely true, and it's hard to do when you don't have any exposure to what's actually. It kind of broke up earlier. It's one of you guys said, I think it was Standing or Maddox or somebody said that there hasn't been any new work in abiogenesis in how long?
5 years.
5 years. Yeah. There's been a ton published.
Maddox, no progress in 5 years.
Yeah.
Let's see, what do we have here? No self-replication, just replication.
Well, it depends on what we're talking about. Are we talking about self-replication of macromolecules or self-replication of cells?
Well, the system at 11th. Did you ever watch that video we did with 11th dimension? I didn't do it. And I did send you the papers, didn't I? I think I uploaded them to our share drive. So
I'll have to check them out.
Yeah, I'll make sure you get that, and you should watch that. And we should all watch that ABio thing again, because we're supposed to do another round here in September, I think.
Well, and these guys, when they talk about it kind of philosophically, that 100 years ago we didn't have the experimental data that we have today. But I would argue, and others have, that you would still, even under that circumstance, you'd still have a good inferential path to conclude life occurred naturally. And the syllogism could be as simple as, at some point in history, there was no life in the universe, as premise 1. Life exists in the universe today, as premise 2, and you conclude life arose naturally. If by definition, if you accept premise 1, even if you have to go all the way back to near the Planck time, if there's literally no life in the universe, then what non-living agency are we talking about that could have kick-started life?
Mm-hmm.
We know which non-living, we know which agent they're talking about, but it's a bit of a contradiction.
The papers have a hypothesis, basically a sort of a self-replicating, self-changing system that dynamically feeds back into itself, as near as I remember. And it was pretty provocative and very, very good papers. So
Yeah, I mean,
So
I think the
11
idea of... Oh, go ahead.
Go ahead. Sorry.
Yeah, I think the idea of like self-replication is like it pertains more to the thought that when you have like some sequence that's undergoing replication, it doesn't seem to be overly significant if the if the final molecule that's produced by this process isn't in some way identical, if not similar, like to the parent molecule that was used, like as its template. Like in some sense, like there should be like some degree of like accuracy in terms of like how some primitive RNA strand is being replicated and reproduced, you know, in mass quantities.
and
When you don't have the when you don't have the protein machinery to help you achieve this level of precision. I think that's kind of what's meant in the field by like self-replication.
Yeah, I think it was. The idea is that natural selection occurs very, very early, and that's part of the hypothesis that it's just Darwinian evolution of very, very small RNAs and small proteins or small peptides and stabilizing peptides. I think it was. We have been so busy in so many different directions. And somebody has to edit that 2-hour monstrosity that we did on Kunin's paper.
Well, you're the only one with the video file.
What's that?
So you're the only one with the video file.
Didn't I send it to you guys?
No.
I think you did send it. I think you said, at least to me, you sent it. I do need to go through, and we were gonna let him edit it, but we were gonna pick out time.
Oh, like the Google Docs of it. Yeah.
Yeah, I set the Google Doc up, but I can also put the video on the shared drive. So we're only using about 4% of that shared drive. So,
well, I just
had it for a month.
Is Standing still in the chat? Have you guys noticed?
No,
I haven't seen Standing in a while. I'm pretty sure he left.
No, he's over. Praise that I am is having they're having a after debate party over there.
Oh, okay. Well, I can talk it through with you guys. So there's a bit of a bone of contention right now between Dan and Standing, centered around these retrotransposons, and I copied. I wrote down kind of what I thought the disagreement was, starting with Dan's comments from when they did the Young Earth Creation Book Club, and then Standing's comments from a couple of days ago where he was responding to that. But in this chat, he acknowledged that he made a comment along the lines that he got the information about jumping genes from this video Rob Carter did.
Right.
I just wanted to point out at one point in the video, Rob Carter is talking about transposons as potentially a way that cells could increase production of certain genes, and that it may have a functional role. It was highly speculative, but he did something that was a little misleading. Dr. Carter did. He as an example, he brought up liver cells. So liver cells can actually make extra copies of entire chromosomes. They can become known as polyploid. Yeah. So he brought it up because it's a great example of how, and it actually kind of illustrates the point that Carter was making. He was drawing a parallel between retrotransposons and these liver cells. And I encourage you guys to check out the video. The way he actually described it, it implied as though in liver cells, retrotransposons were an actual mechanism used by these cells to increase production of detoxifying enzymes, and which immediately got me to searching the literature. And in fact, it's actually a fairly well characterized mechanism, and it's a modified form of cell division where there's extra rounds of S phase where you have additional copying of the genomic DNA, and has absolutely nothing. I have been unable to find any paper where anybody's proposing retrotransposons play any role whatsoever in this phenomenon we see in liver cells. So I just wanted to point out, if any of you guys check out that video, and more importantly, if a guy is speculating about possible functional roles of retrotransposons and he jumps to liver cells, which are not linked to retrotransposons, that's misleading at a minimum.
Mm-hmm.
Yeah, we pulled up the paper that Standing referred to, I think, that Rob Carter referred to, and we have that somewhere in the chat here. I'll put it in the description tomorrow, so we have that paper.
So I read that paper. This was one that Standing shared in his video where they were doing debate prep for Gavin a couple of days ago, and that's the video I was referring to, where Standing responds to Dan's critique of his book. It's a great paper, and it actually, I wish I had been on earlier when you guys were talking to Dan about this. It kind of forces the members of kind of this discussion to get to the root of it. Is it, if retrotransposons through successive generations gain function, does that mean that common ancestry is overturned? I don't think so. I mean, we know the genome is dynamic. We know new genes can arise. We know genes can fall out of use, become pseudogenes, and then become, completely missing from the genome. In that paper, what they were talking about was certain retrotransposons. So there's three major families: ALU, SVA, I think. I can't recall them exactly. This is not my field. But that Nature paper, they basically developed a new technique called RC-seq, and it allowed them to do high throughput screening, meaning they can look at lots and lots of samples really quickly. It's jargon from the field. And they are proposing, so this Nature paper that Standing shared is by no means definitive, but what they're proposing, because they now are able to look at the distribution of these retrotransposon sequences in humans at a resolution that they weren't able to see it before, but they think it actually plays a role in brain development, that you can have different cell types, different neurons forming, and it's at least partially due to the presence of these retrotransposons, which would imply they have a functional role. So not only have they linked it, and they call it a mosaic, where if you were to take a sample of any other tissue in the body except maybe the liver, and you look at the complement of chromosomes within each cell, they should be exactly the same, clones. So this doesn't hold true for gametes. It apparently doesn't hold true for liver cells, and it also apparently doesn't hold true for the brain. But that what they were speculating on is that maybe this is part of why we have the brains that we have, and it would be interesting to look at other organisms with large brains, like other primates.
Yeah,
maybe co-opting retrotransposons, which were present due to ancient viral infections, co-opting them biochemically actually created, a more highly functional brain. And Carter's talking about this. The paper does a much better job. The way Rob Carter presents it is though, basically, hey, all these evolutionists, they always, they've been talking about junk DNA, non-functional DNA. They've pointed at retrotransposons as though they were non-functional junk DNA. And it turns out these things are functional. And I think Carter overplays his hand in it as to exactly how functional these DNA elements are. Can't even call them genes. But because Carter overplays his hand, others, who watch the video are also overplaying their hand. Yeah. Go ahead, Speed.
I'm fighting with Smokey in chat. I asked him to call you a trash clown, Walker, and he started saying, "You just don't get it." And then he said, "Yes, have established the opinion of an irrelevant old degenerate isn't worthy of consideration." And he says something about my character. Our disagreement.
Wait, what about a young degenerate like myself? Does my opinion matter? Can he call me a trash clown? What were you saying though?
I'll say you agree. You agree with me. Let's see. Walker agrees with everything I say.
I would say everything.
Come on now. We got to get you under. Yeah,
we gotta throw out the big guns here.
Yeah. Anyway, he hasn't given me any reason. He claims I have a degenerate character. I'm not entirely sure what he knows about my character. Other than that, I didn't listen to him when he warned me. Warned me not to do a video.
You're going to be on the smoke list?
I was going to be on the smoke list, yeah. Anyway.
He has a pretty cool name for it, though.
Yeah, we didn't go over his... We didn't go over his sexist bullshit. And maybe I could put those comments that were the one that was deleted, and but no, maybe not. Anyway, let's see. Walker might be indoctrinated. He might be very insecure and emotionally weak. And look, that sounds like a trash clown to me.
Well, I mean, we're getting somewhere. But wait, so he just said I'm indoctrinated and emotionally weak?
Yeah, and you look up to the nearest semblance of authority he can cling to. Oh my God, he is he a judgmental person?
I don't know. I mean, I don't know. I think that's a very interesting claim. I haven't heard that I am a suck up to authority before, so that's a new one.
Well, you know what's really cool? I just asked one simple little fucking question. I asked him to call you a trash clown, and he just went on this big diatribe.
Passionate diatribe.
Passionate diatribe. Anyway, continue, guys. I'm sorry. What happened? They're Googling. They're Googling.
The Googling?
They're Googling. I don't know what they're Googling. They're probably Googling trash clowns. No, I don't know what they're Googling. Anyway. Okay, guys, I'm sorry I interrupted you.
Oh, no, you're good.
Okay. I am really tired. What's happening?
Are we saving the Jen Aldrich thing for later then, or what?
I have another Maddoxism.
Okay.
The Jen Aldrich thing, we could do that. Let me see. I gotta do something with this goddamn.
If you don't mind, I mean, if y'all want to do the Jen Aldrich, that's fine. But I wasn't around for the last Maddoxism. I'd like to comment on it. If you want.
Oh, which one? Oh, yeah. You want to see the third one? Okay.
Yeah, if you have one teed up, let's listen to it and comment.
Yeah.
Okay. Oh, let's see here. See if I can get this right. Get her right the first time. Okay, this is Maddoxism number 3, I think, and it's right here, and it should be playing.
Let's go with the second one. So we have, you claim it is nothing but cascade chemical reactions. Well, what we're discovering with structural biology is that things are happening in literally different spatiotemporal regions that are happening simultaneously to another one that are then used in conjunction, but there was no initial cause and effect of the process to occur.
What?
No cause and effect.
Wait, do you have the like transcript of this, right?
I do.
You should read out that transcript because I need that like a word by word breakdown.
Okay, what we're discovering let me go back to the top here. But cascade chemical reactions, what we're discovering with structural biology is that things are happening in literally different spatio-temporal regions that are happening simultaneously to one another, that are then used in conjunction, but there is no initial cause and effect of the process to occur. Okay. This is Maddox speaking in tongues.
Yeah, as per usual, this is mostly a nonsense statement, and those of us who are familiar with the language of molecular biology and speak it fluently, we're left to kind of speculate as to what exactly he's talking about. So some of the elements here, he's got
40 nucleome
cascades of chemical reactions.
Okay.
He's got
ditto.
Spatiotemporal, what was it?
Spatio-temporal
regions or something.
Check.
And he mentioned structural biology. And Speed, you think he's talking about the 4D nucleome?
Yep, because he got that from Cell Cordova. So that's one of their big talking points.
Yeah, so but then he puts the cherry on top and claims that there's no cause and effect explanation for these patterns we're seeing. I'd love to see
I'm still alive.
A legitimate paper making that claim. That's my problem. That's why it's hard to listen to these guys.
Okay,
he's in the field. Go ahead.
He's not done yet.
Oh,
there's more.
Go on.
Okay, so when you look at it that way.
So when you look at that way, you have, from a reality perspective, you have two separate functions being executed simultaneously, but then conjoined to have a alternative outcome. And this is reduced to such a simplistic, it's not simplistic, but such a fundamental component of life, which is the ribosome and the translation of mRNA into a polypeptide, that there is no chemical reaction between
the
codon and the amino acid. It is literally being read. This is a recognized premise in biology. And so
molecular alchemy apparently can't hear him.
That
you can't hear him?
That's unfortunate.
Who can't?
Molecular alchemy couldn't hear him, so he's now rebooting.
Oh,
that's unfortunate because that was gibberish. I'm confident. I'm pretty confident that molecular alchemy is just reading too deep into this, and there is no argument there. There's literally no argument.
Cause and effect of.
Okay, we'll start over if he comes back. He's rebooting. Midnight, did that blow your brain?
Midnight, are you convinced?
Okay, I am officially dumber than I was 10 minutes ago. Thank you.
Okay, we're back where we started. You want me to run that again?
No, I'm gonna kill myself.
Just read it if you would.
Okay, where the hell is it? So when you look at that way, he didn't say at it that way. He said at that way. You have from a reality perspective, you have two separate functions being executed simultaneously, but then conjoined to have an alternative outcome. And this is reduced to such a simplistic. It's not simplistic, but such a fundamental component of life, which is the ribosome and the translation of mRNA into a polypeptide, that there is no chemical reaction between the codon and the amino acid. It is literally being read. This is a recognized premise in biology, in cellular biology, information theory, that there is literally a read of the data being executed by the ribosome. There is no chemical reaction.
All right, speed. I love you, but what the? So
I know what the fuck.
Reading it. Isn't it a chemical that's reading the information?
And he's still not done yet. Yeah, it is. It is a chemical read. So to argue that there is nothing but cause and effect chemistry is absolutely eviscerated at the fundamental of the creation of a protein. There is no chemical reaction between them.
So clearly that's how he's interpreting these papers. The problem is that over and over again, when you follow the paper trail, when you actually check out this guy's sources, that's not what a reasonable person would conclude, in my opinion.
Well, okay, the whole thing about it being a code, we call it a codon, for fuck's sake. How long have we called it a codon? Since 1960? You know? I mean, obviously, everybody accepts that there's a code thing going on. And then it was what, in 1970 that they figured out the tRNA, or that might have even been in the 60s. And so they knew that there was this separation, but he thinks this separation implies that there's no cause and effect, which is pretty dumb.
Well, didn't he get pissy whenever people accused him of word salad?
Oh yes. This is literally speaking in tongues.
I mean,
no, it's worse than that. So imagine you took him up on his $1,000 offer, and he's standing before
oh my god
three or four faculty members who speak these languages fluently and who know how to like we argue with each other all the time. That is what the scientific community does.
Right.
He would be laughed out of the room.
That's like to speed earlier this week. I was like, does Maddox not think he's going to get laughed out of this room? I mean...
Oh, no. He thinks he's going to get a diploma. He's going to get an honorary PhD.
He thinks graduate students don't talk such nonsense. And I've heard graduate students say some stupid things.
Really? Oh, man.
Graduate students are saying the stupid things. I can't even imagine what you think of us lowly undergrads.
Oh, man. If I ever say anything stupid, molecular alchemy, feel free to call it.
Yeah, please tell me if I say something stupid.
Oh my god!
I mean, I'm not even an undergrad, but feel free to correct me. Obviously.
Anyway, yeah, that was a really good Maddoxism, and then we had two others that like one I read. I think one we read together on another morning report, but.
Mm-hmm.
And then we have the whole, there's the whole sexism thing, but I don't know if we have the bandwidth for that tonight.
Yeah, okay.
I think I've lost enough brain cells to conclude that the answer is no.
Anyway, we got to find more Maddoxisms and
the thing is, I'm convinced, as convinced they are of God existing, that Maddox is like the gift that keeps on giving. You can tell him how he makes himself an asshole. And without fail, he will just one-up himself, like without trying. It's truly beautiful. I mean, it's almost an enigma.
Clap, clap, clap.
With the exception of Tommy Hall, have you ever seen something just like so beautifully shitty before?
Tommy Hall. Did you see that debate, Godless Girl and Music Analyst going at it?
Oh man, that is. That's a fair question.
What?
Was
that the one where she was sleeping?
Yeah, it was T-Jump and Godless Girl and Smokey and Music Analyst.
Hey, I just posted this over on Praise.am. I'm pretty sure I'll get a little reaction out of it.
Maddox's thesis.
The $1,000 challenge. Oh, shit.
You should honestly take him up on that. Why haven't you?
Well, the
$1,000 though. I asked Speed. I was like, "What is this? Is he Dr. Evil's nephew? Like $1 million? Like $1,000 to travel across country?
Yes.
Make it $5,000. You know, $1,000 is not.
Why does he
Pocket
want to do it
change,
in person? That's the
That's
strange
what's
part.
creepy. That's where I think he imagines that he's going to get all these accolades, and I don't know.
It's going to be one of those moments, and everybody clapped.
Yeah, exactly.
That man was Albert Einstein.
Yeah.
Look, all of these guys could publish their work if they really had a rigorous, synthesis of the literature. If you put together a high quality opinion piece, an editorial, you can find a mid-tier journal that will publish it. And the fact that he may not have the credentials, may just slow the process down, but it wouldn't prevent it. If these arguments legitimately have merit like he thinks they do, then he needs to get into the discussion, because right now he's on the playground, and not actually in the discussion.
Yeah, well, I told him I can find professors.
Debate's not happening on YouTube. It's just not.
If he put his stuff together, I could send it to PZ Myers, and I could find professors at the U of M, because I have a connection where I could actually put it in front of some professors if he wanted to. But he wanted to do it in person, and then I said, okay, come here. And then he said, no, I don't want to come there because we don't have any police in Minneapolis, he thinks.
His ability to understand politics is precious.
Right. It's really precious.
Let's be glad he didn't ask for an angels in the outfield kind of debate.
Right. Yeah. And then I told him, I said, well, you have to leave your weapon in Nashville. And he said, stupid, I'm gonna fly. You can't take a gun on a plane. Well, yes, you can.
Yes, you can.
Yes, you can. I've done it.
I'm not sure how many times he's traveled, but yes, you can.
Yep, you just check it in baggage and they'll take it.
Wait, so you can get a gun? No, wait, that's not on carry-on. Never mind.
You can't carry it on.
For a second, though, I was thinking, wait, so you can take a gun on a plane, but you can't take your fucking shampoo?
No.
No, they have. It has to be locked, unloaded.
It has to be in a case. The bullets have to be.
Different airlines have very specific rules.
Yeah, you have to have a regular case, and it has to be locked. They have to have the key. A whole bunch of shit like that. So, but they just look at it, they inspect it, and they check it, and they put it in baggage. So, yeah.
I'm not as familiar. I'm not as familiar with these guys as you are, but as you guys are, I'm kind of new on the scene here. But it seems to me like standing seems pretty solid as far as reasoning through these ideas. My critique, if anything, would be that he doesn't have rigor when it comes to evaluating his sources.
Yeah, he just stops at it.
I don't know where Maddox is a different story completely, though. I don't know where he comes up with this stuff. In fact, it reminded me of something I was going to tell you guys. I don't know if you've ever heard of William S. Burroughs or the Beatniks.
Yeah.
Writers. So they had a technique that they would use to kind of stimulate creative writing, and it's called the cut-up technique, where they would take magazines or newspapers and they would cut out words or phrases, and they would kind of mix them up and just let words randomly, look at words in random order and use it to actually influence their writing. And it's a well-established literary technique.
That would explain Allen Ginsberg and Bob Dylan. Ginsberg was an influence of Dylan, so basically, yeah.
In the same way, I actually do find some value in what Maddox says, because he literally strings words together that I would never, I have never come across in 20 years of working in this field.
Yeah,
though somebody cut up scientific journals and organized them randomly.
Well, I got an idea. Maybe you could be one of the professors.
Yeah, we could get molecular alchemy, Dr. Dan.
PZ Myers.
PZ Myers, yeah.
Yeah, and Rob Carter. Just to be fair,
Where does Rob Carter? Does he teach somewhere?
I don't know what the hell he is. He's some sort of AIG. I think he's got credentials. I can look him up on Rational Wiki. Let's see. Or is it?
Yeah, I'd be happy to be part of a panel reviewing. I mean, so I'm on right now. If you're a graduate student working on a PhD, you have effectively you have what is the term they call it? Your committee, the dreaded committee. You know, it's usually 4 people from your department and then 1 person from outside of the department, and they present their work to us periodically, and we decide whether or not they're making progress. We basically hold their entire academic future in our hands.
It's terrifying.
It's Robert Carter, isn't it? Dr. Robert Carter?
Yeah, he works at CMI.
Yeah, but Rational Wiki doesn't even have an entry on him.
Oh, wow.
He's a marine biologist, which really...
He doesn't even make Rational Wiki's list.
Poor guy.
They talk about Rob Carter has, he's a PhD marine biologist, which is solid. The guy's familiar with biology, but just listening to Dan talk about evolutionary biology and the stuff he talks about, and my fields of expertise, having a PhD in marine biology does not qualify you to speak on genetics and population genetics and human health or lineages, evolution. I mean, it just doesn't. And this video that Standing shared, I think is a good example. This guy's, they overplay their hand. Which is not consistent with the community. I mean, we are very careful. We qualify things, and we don't make an assertion in writing that we can't back up with, a stack of historical publications, as well as our own several years worth of experimental data.
Here's the man himself. Is this the one that he?
No, I'm trying to remember the title was something along the lines of the genome is
four-dimensional design
than we imagined.
The four-dimensional design.
No, the title of the video is something along the lines of the genome is far more complicated than we imagined. It was the channel is not Dr. Rob's name. It's like biblical interpretation or it's biblical something is in the name of the channel title. I'll find it real quick. I'll put it in chat, and we can actually the point that Sandy showed started at 7 minutes. Is this it? This may be it.
7
no,
minutes?
This is not it.
Not it. This was him fishing, I think. I don't know. But he's
That's the fundamental heart of that whole system. You have a problem down there, the whole thing breaks down.
And when someone wants to change that, how many people have to think for how long to make a simple change to the basic programming?
Start monkeying with that operating system, with that system software, you have to be very careful.
I think John's been watching this.
Yeah, and that's what we see in life.
But
Huh?
Why is a PhD in marine biology talking about DNA?
I don't know.
Because they only have like 1 or 2 geneticists.
Yeah, Georgia Purdom for God.
Yeah, that's why I said 1 or 2.
Yeah, and she and like Erica points out, they always have them talk about something else that is not their field. Like they'll have Georgia Purdom talking about paleontology when she's I think she's a geneticist or a biochemist.
Yep.
Or
she's
something.
a geneticist.
And then they'll have their biochemist or their paleontologist talking about geology.
Wow. So
Standing's back.
Welcome back,
I've been
Sammy.
in academia like long enough to notice this. Like, there's a, and maybe it's just something I perceived, but molecular, you probably noticed this as well. Like, there's a difference between like the type of language people use, like when they're experts in a field, as opposed to like if they're talking about a field kind of peripherally, if that's the correct word for it.
Yeah, no, I agree. I just put the video in the chat, in the StreamYard chat, standing or speed. My bad.
Where is it? Where is it? Oh, you put it in private? Okay. Let me see if I can get that up. I think, does this work if I just put this video in here? Standing is in the chat again. He's back. Seven?
I like that baseline.
That
very
anything
groovy.
humankind has ever been able to reproduce.
So did we want to go to 7? By the way, Standing, are you still here? If you are, you left your stream unattended last night, and all of us got on there and we were like watching porn for about 2.5 hours. You should probably check that.
So the section Standing shared was at 7 minutes, and again,
Okay.
I mean,
This is
I
this
would just
short?
encourage him to use more rigorous sources. But we
Want me
can
to
talk
run
about
it?
Yeah, it started at 7 minutes.
Conformational changes, but it's more than that, because the 2D interaction network also changes, and it's more than that. The first dimension of the DNA, that linear string, can literally be recoded on the fly. We don't tend to do that with our computer programs. I mean, we've always been experimenting with dynamically reprogramming programs, but it's really complicated and a lot of unexpected things keep happening. But the genome does it routinely. In fact, there's one point in development where your brain is producing millions of neurons a minute. And those neurons are produced in specific areas of the brain, and then they travel through the brain to get to where they're supposed to go.
Not exactly.
Well, how do they know what to do? Why is one neuron different from another?
grow.
Well, it turns out there's these little things called jumping genes or retrotransposons that they used to say all retrotransposons are remnants of ancient viral infections. They're just junk DNA. They're just sitting there. They're parasitic. Well, actually, no. The brain cells have actually different genomes. The retrotransposons will pop out, go somewhere else, and stick themselves somewhere else in the genome of the brain cell. And that changes the code, and that changes the type of brain cell. Now we have type 1, type 2, type 3, type 4, because of these little retrotransposons. They discovered that in the mouse embryo. If you deactivate one class of these things, the mouse embryo will develop and then stop. Because a lot of retrotransposons carry the start sequence for different genes. And so then by them popping around and moving in the genome, they change the first dimension of the genome, which affects everything else downstream.
No. I've got a book on neurodevelopment. I can check this out, and I would like to see the source papers for this claim, because no, they don't quite jump around. No.
Well, I mean, jumping gene is kind of the slang for retrotransposons, and Standing provided, a supporting paper. It was the one I was discussing earlier, RC-seq, where they're looking at these mosaic neurons. There may be biochemistry there. The way Rob Carter is describing this stuff, specifically, he said these retrotransposons contain promoters of other genes. I mean, he almost verbatim said that, and I don't know of anywhere where you can support that specific claim.
Yeah, he said they're hopping around. If they hopped around, retrotransposons don't hop around with any prescribed precision, and for the brain,
and it's a rare event. It's not like and Dan and the others were talking about. This is what I would love to see Standing and Dan. I wouldn't mind participating as well. Clarify this, because the way that Rob is describing it here, I could see how Standing got the impression that these jumping genes are functioning almost in a parallel way to transcription factors. which transcription factors literally jump around the genome, bind to promoters, and turn on genes. That's not what is actually being seen with retrotransposons. That in a normal somatic cell, so meaning a non-gamete cell in the body, just the average human cell, these retrotransposons play probably very little or zero biological function at all. But there does seem to be, like in the brain, like in the placenta, there does seem to be some functionality, and we can get into the actual data, how they determine that, what are the implications. But again, like I said to you guys earlier, what we need to clarify is that do we all agree that these retrotransposons are the result of ancient viral infections? That's number
Yes.
one.
Well, hey, hang on a second. He's accusing me of not looking at the data. Okay, a retrotransposon occasionally will jump to another spot. It basically it gets activated. It inserts itself in another spot in the genome, and that either becomes part of the germ line, which is a rare event, or the somatic line. Okay, in order to work in the brain to be functional and actually have something to do with the wiring of the brain. And what happens with neurons is there's a neurogenesis area where the neuron cell is created, and then there is a gradient and a set of a set of cells that develop around it. Okay, and it follows these paths all the way up to the cortex. Now that is a very prescribed route. It has to happen exactly the same in every organism. So that is the result. And it may, in the mouse, they may have found some retrotransposon that in the past had jumped to that spot and then been co-opted as part of that developmental process. But it does not jump while it is in development. If it did that, every brain would develop in a radically different method. That is not possible. Brains are very, very prescribed.
There
Correct?
Two things. It may be a combination of things, so that you have plasticity due to elements like these co-opted retrotransposons, but you also have rigidity in the development of the brain, so that you end up with individual offspring that are mostly alike the other. So there may be an equilibrium there. But the most important part, I think, is the co-opting side. That these are potentially rare events and not necessarily a defeater for common ancestry.
Okay, he's claiming multiple transposons jumping around during development. Is that what you're saying, Standing? Okay.
I think the first thing to get on the same page about is whether Standing thinks retrotransposons, we're talking about a specific feature of genomes in modern organisms like mammals, like humans, whether Standing agrees that they're artifacts from ancient viral infections. I think that's the first part, and I think he'll agree with that. The way that we concluded that retrotransposons are viral in origin and not de novo genes, endogenous genes that are human just as much as everything else around them, is because retrotransposons contain an actual viral gene, and they produce a viral reverse transcriptase. Like that's how they have high homology. The promoter of retrotransposon, the actual open reading frame, the flanking repeat, there's high homology with viral genes compared to other human genes.
Was it Bork, or was it
the other thing? I can share the link to that Nature paper. You can search for retrotransposons and RC-seq, SEQ, because that was kind of the two main elements here.
Here we go. Survey of transposable elements. That one. Element classification systems, fundamental update. Okay. I'm trying to find today. We got X. We got, let's see. I'm trying to figure out where it is. I think it was Bork. Yeah, 10. No, that's 10 things you should know about transposons. It's not it. So what did I do with the paper? I just downloaded it on one of these tabs.
So the other thing I was going to say, and I wanted to point it out to Standing, like this is a recent paper, and even this stuff related to placenta, like this is, discoveries go through kind of developmental stages all of their own. You know, you have the initial discovery, which may, you know, raise the hackles of the establishment because it contradicts something. Then you have reproduction of that discovery by other labs, which raises the amount of respect it has in the field. And then you know, there's this fairly well-defined process where these paradigms shift. I would say this Nature paper, these neuron mosaic, the role of retrotransposons in placenta, this stuff is still fairly new. You know, this is, if you're looking at retrotransposons as though they are a defeater of evolution, you're effectively looking at a few dozen papers versus hundreds of thousands of papers. And I think this is the right paper. It's Standing still in the chat. He can verify.
I'm pretty sure it's still here.
Scroll down. Let's look at some of the figures. I don't remember. So this is an important part of this, is that this is a technology innovation paper in as much as it's a paper reporting about biology. So part of the reason these guys got into Nature, which is, you know, the epitome of scientific journals, is because they developed this technique. So everything else aside, this is kind of a report on technology development. And then they go on, because you have to do this in a Nature paper, you have to show if you're producing, and this is something that our lab does. If I drop the names of some assays, you know exactly what lab I work with. So I won't do that. But basically, you talk about what your new technology is, and then you apply it to a biological system, and you come up with a new biological discovery, which these guys, you know, seem to have done with this potential putative role of retrotransposons in neurological development. And they go on, they actually talk about further evidence supporting this idea that retrotransposons may have a functional role in the brain in this context, is that they're linked to certain neurological disorders. So there's ancillary papers that they cite that kind of further support these unique and kind of exotic DNA elements actually being necessary, maybe. But they're not establishing that these particular type of retrotransposons are necessary for proper brain development. But they're laying the foundation. It's definitely interesting stuff. But again, I don't think it is the smoking gun, you know, that refutes evolution that others may think it is. I think I think it's reasonable to conclude that retrotransposons can be co-opted through successive generations.
Yeah, they validated that. Basically, it validated that there was a long terminal repeat insertion here. So they're validating that it happened somewhere in the past, basically in the genome. They're not claiming that it's jumping around now the way everybody, including Rob Carter, is implying. I mean, these things can't jump around while the organism is developing a brain. That would result in death.
Yeah, honestly, if it was as dramatic as Rob Carter was saying, like when you have genomic instability, and if we're talking about jumping DNA elements, we're talking about genomic instability. That's one of the 12 hallmarks of cancer. Like, it's unlikely that we're going to find, and we've talked about this, epigenetics, 15, 20 years ago, epigenetics revolutionized molecular biology, medical research. Everybody was looking at their areas of expertise in a new light. I don't think we're going to have a similar revolution with retrotransposons, and I certainly don't think it's going to overthrow common ancestry.
Yeah. Yeah, I have to look at this in relation to development of the brain, and I do have a book on how the brain develops. So, of course, I do.
Hey, Standing, if you're still in the chat,
Well,
I'd love to talk to you.
Yeah, come on in, Standing.
Well,
anyway,
if Standing comes in, I really want to talk to him, but I'm getting tired. I probably need to go.
Hey, I got a question. I have a question, a general question. Who has kids that are still up at midnight?
My 5-year-old is jumping around
really?
right now. Yeah.
Wow. Coffee?
You want to say hello?
Hello.
Hello.
I also want to learn about your bodies.
Oh, good.
Brains help you think about stuff. And I don't want to say the bladder, but I already said it. The bladder holds all your potty and poop.
So my great niece, she was 5 years old, and she wanted me to buy her books. She loves looking at the wiki. She wants to know what's inside her body. So I bought her anatomy coloring books, and then a kid's encyclopedia of the body. And she carried it around everywhere with her. And we figured out she's either going to be a doctor or a serial killer. We're not sure which. She's a strange little kid. And she didn't talk until she talked with, she used expressions of her face, and she would point at her face to emphasize, but she didn't speak until she was like 3.5, something like that. And she is just an incredibly different child, but really interesting. So, but I bought her a really neat encyclopedia of if was that little girl molecular?
What's that?
She's a little girl.
Yeah.
Okay. I have a really good book. It's basically about the body, and it's for children. It's really well illustrated, and I think I can find. Yeah, I could say I'll try to remember to send you. Right? Yeah, because my great niece absolutely loved it. So anyway.
Yeah, she's really gotten into human anatomy and physiology.
For her birthday,
I got her this little model that has it's kind of an educational tool, but it's got like all the major organ systems in the thorax.
God,
she loves that.
She really needs to meet my niece. Those two would have a good conversation.
Molecular Alchemy, Standing for Truth, wants me to let you know that he's been buttering you up in the comments for a potential discussion. He's been like complimenting you and stuff.
Okay, cool.
Yeah.
Yeah, we can set that up.
He's mostly been agreeing with what you were saying on like the retrotransposons. He agrees that it's not a smoking gun, that kind of stuff. But he thinks there's probably more to it than we know, that kind of stuff.
Yeah, I'd love to talk to him.
Yeah.
I've watched a few of his debates, and I can tell that he gets excited about the science in the same way that I get excited. And I don't know if he's still listening, but I can say that passion, that motivation was enough to propel me through undergrad, graduate school, that if there are some really lucrative jobs out there. If you're really excited about this kind of stuff, like, get into it.
Get into it, yeah.
Yep, and you can probably tell them what else happened to you when you started studying it.
At a more than
Yeah.
surface deep level.
That's another station.
Yep.
Are
Okay,
we wrapping
guys.
it up? Because I've got to put this to bed.
Yeah,
Yeah,
I
we
think
gotta-
we're all getting kind of.
We got to wrap it up. We're Immutable just left.
Yeah, he had to bounce. I mean, he said goodbye in the private chat, but he didn't want to interrupt the conversation.
Okay.
Gotta watch the
Okay,
chat
guys.
more. Nah,
Huh?
said you gotta watch the chat more.
Oh no, Leo's here.
All the cool
We're
stuff
just about to leave, Leo.
I knew
We're
that was
literally
gonna
saying
happen.
bye.
The minute I joined, I'm like, they're probably at the end right now.
Yeah,
I
we're
spent too much
totally
time at Steve's rum and coke night, so sorry about that.
we're totally
Okay.
burnt out.
Yeah, how long you guys been going for? A couple hours at least?
2 hours, 49
3
minutes.
hours.
Probably our
3
longest
hours.
live stream.
3 hours. Oh wow.
Jesus
No,
Christ.
2 hours and 50 minutes.
I see.
Probably the longest one.
Standing for Truth has just been blowing up your side chat.
Yeah.
Yeah, he's been off and on in this chat.
He's learning. The guy is learning.
I
Hope
can't
so.
see that.
I hope that's what we can classify it as. Learning. I really do. So guys, actually, you know what? I'll ask you guys later.
Okay.
Okay, you want to go on live?
I mean, I
So-
can ask it live. It's just I was
No,
kinda
we'll
not
end.
really
Well,
relevant.
we were ending it anyways.
Oh.
Yeah,
Literally as soon as you
we were
can.
going to end anyway. So, okay, ending broadcast. Goodbye.
Say goodbye,
Anybody
Leo.
who's
Hi
still
guys.
listening,
Bye guys.
stick around, Leo.